You know, I forget what movie it is, but there's a wonderful quote that says, "Don't you love it when you go to the bathroom at a restaurant and you come back and your food's on the table?" The equivalent of that is, don't you love it when you go to the bathroom and you come back to the ROV control room and the ROV's on the bottom? And just
looking at rocks. Rocks with sponges and several Okeanos, so we're sitting down in a nice place, and even a sea pen in the sediment so that we can do an early comparison of our sediment-dwelling corals and our rock corals. So for folks on shore, we're just getting settled. Pilots take a little moment to... oh, we got the holes right in front of us.
No.
Right on the holes.
No.
This time we're suctioning those holes.
Oh my goodness, there are the holes.
Right by the lasers. Good eye, Anna. This was our video engineer, Anna Sagatov, next to me, pointed at these holes rather excitedly from one of the cameras that you're not seeing. We have a whole series of cameras on this ROV, but you can't see them very well, but there are 4, 5, or 6 in different directions here, so I wonder if Mike Vecchione's on the line.
After that, I have a bailout southwest heading 210.
So we have a number of things to do before we get started. It looks like maybe we're even at a point where we're going to have a pilot turnover. I'm seeing a lot of chatter going on in the front row. We usually get a sit rep as one pilot and navigator and co-pilot comes on. They're updated on the total situation, how are the vehicles responding, what has been the science discussion, etc. So we will let that play out. I'm not sure if the camera balance has been done, the white balance, so we may have to do that. But then we'll get on with our dive, have a look at our fauna. But there's been a lot of excitement about these holes that we saw. Actually, I don't even recall what dive that was for. Our EC is here, Derek Sowers, expedition coordinator, and he's been paying a lot of attention to all the interest in these holes as well. So dive 4, we saw these linear progressions of holes, and here I'm seeing some linear. A lot of them are actually curved and over one another, so this is kind of interesting. And this is considerably shallower than the ones that we saw, which were at about 2,500 meters. And in the paper that Mike Vecchione and his colleague wrote, they were at about 2,000 meters, I believe. So we'll have a look at these ones. And Mike, it's my opinion that if we see any of these with really fresh holes, that we should suction in them after we get a good look.
ROV, when you have a chance, can you turn the lasers on, please?
They are on. They're right next. Right there.
Oh.
Yep.
Thank you.
Copy that, pilot. Yep.
You know, we do some wonderful, beautiful exploration, both in terms of the geology and the biology, and it's unclear to me what category these holes fit into, but it's clear that everybody does love a mystery, the public and scientists including. So as the public has been going on around and hypothesizing about what might have caused these holes in social media put up by our wonderful partners at the Global Foundation for Ocean Exploration, scientists have also been chatting back and forth about possible mechanisms to create these holes, and we've heard everything from deep ocean floats to gastropods.
I had an interesting email that suggested that something had laid some eggs subsurface, and when those eggs hatch, basically they create those eruptive mounds. Whales feeding in the bottom, beaked whales specifically that have a little bit of a quadrate sides to the beak, and they probe it into the sediment. But of course, one of the things we're all interested in is how almost perfectly aligned these holes are, and the regularity of the spacing of the holes and the size of the holes themselves.
And the shape of the holes. I will still say the angularity, the rectangularness.
Right, I agree. I mean, my first thought also is that looks something man-made. But as Mike Vecchione has pointed out, and they did plenty of research when they were publishing their papers, if you look in the fossil record, there are also indications of similar sorts of features. And so this suggests that there's an organism that has been making these sorts of features for a very long time down here in the bottom of the ocean.
Yeah, and that's important to appreciate. We've just seen these over the last week and just kind of are, espousing kind of our hypothesis. But Mike's been thinking about this for a long time. He's been observing these for, 20 years? What was the first paper?
The first paper was just this year, but the observations that he had made were in 2004.
2004,
and he referenced, I think, the Heason and Hollister deep ocean photographs I mentioned to you earlier in this cruise, that there was some indication that there was something like it, I think, and that was published in the 1970s. We wondered if we might see some. I did not expect to see some right under the ROV skids.
No, I don't know if it's a blessing or a curse. The ROV team's precision landing right on them.
Yep.
And so, I don't expect to see any young pillow basalts or anything like that. We're on pretty old crust, as I said, about 13.5 to 14 million years old. So we're going to see a lot of pervasive sediment, marine detritus. We're going to see a lot of that carbonate crust that we saw on the previous dive that we got a sample of, which looks like dissolved and remineralized calcite or calcium carbonate, with maybe some dissolution structure in there, kind of dissolving the shells that were actually forming part of it. Interesting, when we grabbed it yesterday, I thought maybe we would see something called fossiliferous limestone, where you can actually still see some of the structure of the shells, but we all we brought it up and we took a look at it. I could not see any shell actual structure in it.
I agree. I didn't see anything that I would have interpreted as a fossil.
Yeah.
There are plenty of nooks and crannies in there. You pointed out that you could see all the way through the rock at one point.
Yeah.
So it was kind of an interesting thing and a wonderful surface for biology. We got a lot of interesting, very small samples. One of the benefits of bringing up rocks is that you have the opportunity to sample organisms that are so small they'd be either overlooked by the ROV or we don't have the tools to pick them up. And so I was scraping off various sponges and tunicates and tube worms and so on.
So out of curiosity, off of that one rock sample, how many biological specimens did you collect?
So the ones that I was able to take off and preserve, I got 4 sponges, a tunicate, polychaete, a brittle star, a very small hydrocoral, and I believe I'm leaving at least one out. And then there were some other things that I'll put in the category of probably single-celled organisms, things like foraminifera, a mucus binding up sediment that may have a biofilm or microbial action associated with it, but not something that I could identify.
So that's at least 8 kind of macro specimens taken off that one rock. That's
off that one rock, preserved, and some of them in beautiful condition and kind of excited. And one of the things I would love to be able to get is feedback later on what exactly was that? Is that something that grows into one of these larger things that we see, or is this a whole set of species that is very small?
And do you think that hydrocoral, was that the one that Chris was originally interested in that we didn't have time to?
It was Asako Matsumoto who was pointing out that we probably should get a collection of that, and yes, I absolutely believe it was the same
It wasn't
one.
Chris because it wasn't a star.
Correct.
Yeah.
Oh, well, we did get a star.
But we did get a star.
Rock,
yeah.
But we got it because we moved the rock.
Yep. And,
exactly.
And you got that, there was the star through this, that was the star through the slurp sample, or that was a?
We got that in the slurp sample.
Yep. And that came up
in beautiful condition.
Yep. And how big was that in terms of?
That was about 2.5 inches wide.
Oh, wow.
Also, the slurp sample, we picked up the rock pin, speaking of things on rocks.
Yeah.
And that came up in terrific condition. And it's really interesting if you compare, what we see in these videos to the size. So that whole colony was only 12 centimeters in length.
Yeah.
So pretty small. Really nice condition.
Yeah, and it goes back to what we talked about briefly on the dive down in terms of this really beautiful relationship between the geology and the biology.
Agreed.
Yeah. And so just preemptively, let me just say, you're welcome.
Yeah, thank you for the rocks that you're going to collect today.
Yeah. Now you can see all that dark black coating that's on that carbonate crust that we see. Originally I was thinking that was primarily iron manganese precipitation on that, but when we got a good piece of that back into the lab, I think it's actually kind of a mixture of degraded and biogenic ooze and decaying of some of that organic organisms, that life. It scrubbed off really easily. There wasn't actually any really hard precipitation onto that carbonate, and that's
interesting.
That's probably, if I had to assume, and I don't know this for sure, it's probably just a function of the slow precipitation rate of iron manganese out of the water versus the much larger deposition rate of calcareous sediment.
So I shouldn't just be saying every time I see something black on the seafloor that it's manganese encrusted, is what you're telling me.
It's usually a safe bet.
Yeah.
But yeah, I think in this case it's a mix. Of for a lack of a better word, just kind of ooze. Yeah. Yeah, I guess we can think about, when one of those, like when one of those bamboo corals dies and falls over, right? It's got that internal calcite structure, right? But it's got that exterior organic layer,
sure,
right? And that's
going to attract bacteria,
yep,
decomposition.
Yep.
One of the things we never. Yep. Go ahead, Copilot. That is correct, yeah. Before we move, we'd like to take a Niskin sample. In fact, we don't want to move at all from our present position. Thank you. Water sample number 2 collected. Thank you. So pilots, you probably heard there's an awful lot of excitement about these lines of holes in the substrate. And what we're looking for, well, we want to just, first let's survey it well. And then what we would like to see is if this line of holes, if we can tell that there's a fresher end, and if we see some holes that look like they're quite fresh, and by that I mean they're not infilled with some of the detritus that Ashton was just describing or settling from the water column, then we'd like to do a suction in it.
ROV, do we have the ability to do a near downward looking kind of photo grid survey of a small patch of this area, or? Okay.
So this particular one looks more curved than some of the others. The laser, so are the lasers back on, pilot, or are they just off frame? Yeah, they were there. Thank you. Yep.
And again, you kind of just see how sharp and rectangular some of those holes are, and then it looks like, they kind of fall, the surrounding sediment falls into them,
right,
rounds them out.
So I think, there's a couple of things here. The fact that they're collapsing these ones says that they are relatively old, I think. And there's probably also a difference on the nature of the sediment, right, about how easily they would collapse versus how easily they would keep their shape. I don't really like either end because they look a little bit degraded, but this one looks a little sharper to me. Would you agree, Ashton? Maybe we could zoom in on 1, 2, 3, like the fourth one down, the second one up from the skid or the bottom of the screen. I'm just trying to look at a fresh one that has nice sharp sides. Let's get a good zoom inside there. I'm seeing a number of other tracks also in the sediment that tells me there are other organisms that are moving about the top of the sediment surface.
And I'm really still hoping that I'll hear from either a member of the scientific community or a member of the public with an explanation that is just spot on,
perfect,
incontrovertible.
Can't be disputed.
Yeah, it just makes complete sense because we've had a number of them, and they all, well, not all, but a lot of them have strong merits to them, but none of them are what I would say conclusive.
Right. A little bit of weakness in various parts of them. Okay, these ones don't seem fresh enough to me to really give us that valuable information. So let's just sort of scan this whole area, right? See what we see.
Some of those middle ones looked pretty sharp, but that's right. We don't know anything about these, so we're already starting with the assumptions that we think that it's making it in a linear track, but we don't know whether that it's starting at one end and going another. We don't know whether it could be
two of them meeting together in the middle.
Or starting in the middle, moving out. So.
One of the things that was discussed in the paper that Mike Vecchione and his colleague write, I think it's Bergstad, and I apologize to the co-author. I really need to call that up so I don't keep saying just co-author. Was that in the fossil record, these holes were connected subsurface by a linear line. Maybe that's Mike calling in now because he can describe it much better than I can if it is.
Yeah, I decided I couldn't stand it anymore.
Yeah,
there you go.
Anymore, and I would go ahead and call in.
Go for it, Mike. So we're going to turn this over to you. Please.
Well, I don't think I have anything to say that you haven't already said, except that it's remarkable that you landed on them today.
We're just that good.
I've seen a whole lot of guesses as to what they may be, and actually some of them parallel what I actually think is going on here, which is I suspect this is something burrowing under the surface and periodically poking up, perhaps to get fresh water into the burrow as they make it longer and longer. So if we could find any kind of evidence of what's doing this, it would be really interesting. Now, some people have commented on the fact that the fresh ones have such well-defined sides around the holes, and I think that is an important characteristic also. But when we were talking about this the other day on the chat room, somebody, it might have been Les Watling, suggested that Alphaeid shrimps make openings to burrows that look very much like that. And so I Googled the shape of Alphaeid shrimp burrows, and I agree with them. Some of them have very well-defined sides where they poke out through the sediment. But, of course, their burrow doesn't look anything like this. But I think that something under the surface that's burrowing and occasionally or periodically poking up to the surface makes a lot of sense. And there are a lot of burrowing animals in fauna out there that make traces involving holes that poke up through the bottom to the surface. We've seen these rings of holes, for instance, which I think also we don't know what makes them. But there's things like spoonworm tracks where there's a hole and then there's several traces in a spoke-like pattern around them. So,
I know one of the things they do in shallow water with. Sorry, Mike. One of the things they do in shallow water with the thalassinacid shrimp, they will pour a matrix down into the burrow to harden and see what the burrow system's like. And I think Ashton suggested that wouldn't that be a great idea? Be able to have a subsea latex that we could inject into the hole to see if they are connected subsurface.
Yeah. Do you happen to have any latex in the ROV's back pocket?
Not today. We weren't anticipating this.
But Mike, do you think it's worth to try and take a slurp sample in a hole if we can?
I think it would be a good idea to try to suction up probably whatever you see as the freshest end of one of those lines of holes. That would be a good thing. Well, you're seeing some nice fish right now.
Yep.
And also there was a big shrimp as well.
We settled on a good location.
Yeah, I don't know if it would be successful, but I think it'd be worth trying.
Yep. I was sort of kicking myself for not suctioning it the other day. I didn't realize the interest this was going to generate, and we've been given another opportunity. So
yeah,
we got to do it.
Poseidon provided you another possibility.
Right. So we got to find the
and it turns out these these are very common in some places. So it's not just a mystery. It's a mystery that's probably important, at least locally important, to benthic ecology in the area.
Yep. Okay.
One of the important things about animals that live down in the sediment is how much oxygen is in the water that seeps down to the sediment, and this perforation would increase the flow of water and the potential penetration of oxygen down into the sediment that would help out other animals or microbes.
Yeah, that's an interesting comment, and we do have CTD measurements of dissolved oxygen in the water, and so we could do a comparison of the oxygenation of the water at the places where we're observing these and where we're not observing these. I don't know if it would lend any insight, but it is another environmental control that at least we have. And we're looking at.
I suspected where we're seeing these. Sorry, go ahead.
Oh, I was going to say we're now looking at another camera, and we can see what looks like maybe some fresher ones. So maybe these would be a good one to try, Scott.
Yeah, that's what we're thinking, and it looks like this end that's above the lasers looks to be a little bit fresher than the other end. The pilot was noting that it seemed to fade over there, so. Yeah, and so there's a pile of something above that track, and then another burrow beyond it, and that burrow beyond it looks a different shape. I agree with you. So one above the laser or one below the laser? That's the question. Right, I think that's something that's just has drifted in and is sitting on top of a hole. Right.
Yeah.
When we had. Yep.
Yeah.
Okay. Yeah, the thing that's just above the laser, I think I saw. Yeah, I think I saw a cnidarian on it, a polyp on it, and I believe that's something that's sort of drifted in. So it's not part of the hole. Yeah, that thing. Exactly. Yep. Okay. I can't believe that I just said don't collect that cnidarian.
I know. I want
this is what the holes will do to people.
I want there to be a completely geological explanation for it. Like there's I'm just expounding wildly, but a subsurface crack in the crystalline basement, and then there's a fluid flow through it, and the spacing of the holes just so happens to be on the frequency of the wavelength of the fluid flowing through it. But that's me just trying to feel like I'm part of the conversation.
Yeah, it would have to be something that occurs in many places.
Mm-hmm.
And we haven't seen any indication of either fluid flow or bubbles. Bubbles would be another thing that could do that sort of thing. But they wouldn't create these kind of holes, I don't think. The other biological explanation is something that's going along above the surface and then poking down, perhaps feeding, like a shorebird does, poking its bill down periodically to try and catch things under the surface. But I don't see any indication of tracks alongside these holes, or Sulak suggested it might be a skate or something like that, which could swim along just above the bottom and not leave tracks. So it's still a possibility, but I think I'm going with the idea that there's probably a tunnel connecting these underneath the surface.
Yeah, because the area around it,
that tunnel and periodically poking them up.
The area around it is otherwise so clean that it's hard to picture something moving above the surface that could be so perfectly aligned and not disturb any of the other sediments. So I tend to agree with you, Mike. Unless it was a float. Bobbing, I can see that, but they're so perfectly spaced. I mean, why would it always be that spacing?
I said it sarcastically the other day, but I think, I'm now really interested whether we could build a device that does, sub-seafloor casting,
Pogo sticking like this?
Well, no. I mean, like, can we build a device that does like a plaster
gotcha.
injection into that? Because we
Sure.
do that terrestrial to model burrows of insects and small animals. You've seen those
Sure.
casts of ants and things like that. But I'm not aware of anyone who's tried it in the deep sea.
Right. Just while we're getting set here.
The problem.
Go ahead, Mike.
Another problem with a pogo stick kind of thing is that these are not round; they are elongate, and the long dimension is always lined up with the direction of the hole, of the line of holes. So if something, perhaps a man-made float or something, was pogo sticking along, it would have to do it without any rotation at all, and it would have to come clear out and then go back down pretty hard, I think, to make the next hole. A lot of ifs involved in that explanation.
Yeah, the pogo sticking of the float. To me, it would be that it's got kind of a spade-like end, and that might align with current direction. But that's actually, I think, the antenna. So it would actually have to be inverted from the float. From the pictures of the floats, I've seen how they float through the water column. So it would actually have to turn upside down, maybe, and then poke its antenna in, which is a little harder for me to understand how it would possibly do that. So
we're ready. Watching. Okay. Yep. Great. Yep. You can see how loose that sediment is. And here on board, we're also watching a canister, and we're seeing that fill up now like a little dust storm. Okay. Is that the chunk that was sitting on top? Yeah, that looks like an old coral skeleton. Yeah, before you go back in, if it clears, I don't know how long that'll take to clear, maybe we could just have a look inside there. No, inside the hole that you just excavated. I mean, I did see it collapse a little bit, so that may not be fruitful, but...
And Ken Sulak, one of our shore-based scientists, is saying that one of the surprises of the previous dives during this mission has been the almost absence of the holosaurs and Synaphobranchid eel, two normally dominant fish taxa, but both we've seen already in today's dive.
So you can see how fine the sediments are here. They're creating this very light cloud that's hard to see through. And it's also sticky. You see how sticky this material is, and I understand that's because of the size of the particles in part, how closely they're packed together there.
And so
but that could explain how steep the size.
Oh wow! Look at that. Yeah.
The sediment.
Yeah. So I would not go to canister one, but let's hold off on sucking anything up just for a moment. I don't want to burn all the canisters on sediment. Thanks. Yeah. Go ahead, Mike.
Oh, nothing. I was just watching the side collapse.
Oh, timber, you say, yeah, yep. Yeah, just that short suction there. We filled one of the canisters, and I don't recall ever filling a canister. That's pretty impressive.
Yeah, I think it's going to be difficult to answer the question this way. I think it's going to require just luck and seeing something poke up when it happens. But, maybe we will.
It's a good question, and I wasn't even thinking it was a sample. I haven't labeled it myself. I think we now have the ability, we have a geology pulled down, so let's just call it geology sample.
Yeah, Mike, I definitely appreciate it. It's going to take a bit of luck, but I would argue we've been lucky already in that we dove right on one. So let's, we're not on land. We can't buy any Powerball tickets. But we've got our own geological, biological lottery right here.
I'm certainly not seeing anything move out of this area where we just suctioned. There's probably not as... Oh, it's... I see. Okay. Yep.
And the other assumption we're making is, we say these look fresh, but we don't have any real time process or time scale in terms of what fresh is. Is fresh something that was in place a month ago, a day ago?
Yeah, I've noticed that some of these lines of holes look pretty fresh at one end, but at the other end, the holes are already filling in with sediment, phytodetritus, and stuff.
Yeah.
It indicates it might have taken a long time to make that line.
Yeah. Yeah, it's a very good point. Yeah, we don't know the time scale of the process to generate that.
I think I want to look again at canister 1 if you could. Just see how much is settled. See what the layer is down there. Thanks.
And we've got a good waypoint fix on where we are now in terms of position. Perfect.
Yeah. Okay. Yeah. So if you're not comfortable putting any more in canister 1, then I would say let's go to canister 2 and try one more at that. So wait, let's look at that hole above on a zoom. I want to make sure that's not a different kind of hole. When I first got up here, that looked like it was much smaller diameter. It wasn't square. Yeah, maybe it is. Yeah, maybe it's just so far. Or it has the same, otherwise the same morphology.
Yeah, and it looks like the sediment's pushed out from underneath.
Yep. Yeah, just a little bit more, but in that hole. Thank you.
Now, what would be really great is if we could find one that's in the depth range of the drop cam that our Azores colleagues use, because then we could drop a waypoint and maybe have them come back to it and look at kind of time scale processes of how long it takes for these holes to disappear, and that we can then kind of back out maybe a weathering rate or a redeposition rate.
I think all of the observations that I've been able to accumulate have been in the 1,500 to 2,500 meter depth range.
Okay, so that's right where we're in right now at 1,500.
It would be nice.
I like that angle, pilot. That's good.
Watch one end of these things.
Mm-hmm.
Yeah. Yeah, you don't need to go any further forward. Yeah. Yeah. It's such fine particles.
You have no idea how deep you have to go.
Yeah, we're here. Let's go for it. In for a penny, in for a pound. Yep.
Yeah.
Yep.
There you go.
I don't think I would recommend using any more canisters.
Yep,
Yeah,
I
I
agree,
think
Mike.
this is
Yeah, this will be the last one.
And just Scott, for your own awareness, I will probably only take one or maybe at maximum two samples of the coral, and those can go in the rock box that is open. So if you want to use the rock box with a lid, I know that's not ideal, but
Yep, thank you.
open for any larger samples.
Copy that.
I was going to say that suction sampler's been through a lot.
I don't want to hog the dive, but I think, rather than messing around much longer on this, if we just keep an eye on these things when they're around and look for any kind of activity that involves them, that would be the best
Will do, Mike. Yep.
approach at this point.
Yeah, I think this is time that's been well spent. There's been a big interest both from the public of it and also just the scientific inquiry. So this is exploration definition.
And who knows what we'll get in that sediment sample. Copy that.
Yeah, I don't envy you tonight digging through all that muck. It's in the
I don't envy Scott either.
Well,
digging
it's
through all that
okay. I
muck.
recorded as a geology sample, so it's Ashton's problem.
No. Yeah, too bad there are no sedimentary geologists at Wash Lake right now. But, you know, we don't have a sieve for the sediment, but we do have some nice sponges I can strain things through.
I'm sorry, some sponge?
I said I don't have a sieve for the sediment, but I do have a nice sponge I can strain things through. I would never.
Ken
Well,
Sulak
save all that sediment because apparently that's valuable for cosmetic purposes.
Oh, there you go.
I was on a cruise once where people were collecting deep sea sediment. Some people were using it to make pottery, and other people were keeping it for cosmetics.
Yeah, so you know, as that those calcareous tests and that marine detritus weathers out, you get an increasing concentration of also clay in it, and so that clay is useful. The clay is useful as a pottery product, but then you know the glass richness of that sediment is also useful for glazes and potteries.
Well, stoke the mystery a little bit more.
Well, thank you for trying.
Oh no, thank you, Michael, for chiming in and yeah.
Yep. Clay.
I think I'm going to hang up at this point and just go back to the chat room.
All right.
All right. Thanks, Mike. Maybe we'll get a cephalopod later.
All right. I'm looking forward to it. There goes a bristlемouth.
Yeah, there's plenty to see around here.
All right. Bye.
See you later, Mike. This is Mike Vecchione from the NOAA National Systematics Lab at the National Museum of Natural History, the Smithsonian Institution in Washington, D.C. So we're seeing a curly whip black coral. I see a couple of Metallogorgia. So pilot, the large white structure in the center, a sponge, and then the fans just to its left. I'm interested to see if that's a two-headed fan or if that's two separate fans. Thank you. It should be two separate ones, but it kind of looks like it's a two-headed. It's a... Correct. Exactly. So this is a Metallogorgia melanotrichos, and I'm wondering if I'm saying two-headed. It actually just looks like it hasn't dropped those lower branches yet, and I certainly haven't seen this often, if I've seen it at all. Yeah, look at that. How interesting. What would be even more interesting is if there were two ophiuroids on it. Those are the brittle stars that are living in close association with the Metallogorgia. In front of the sponge, I'm pretty sure that's a bryozoan colony, that little fan thing. This is probably a Faria glass sponge, is going to be my best guess here. And I will do a quick lookup using the benthic animal guide that's been developed from all these different explorations of the Okeanos Explorer, and we try to take out the best images that we can, and when we have samples we send them to experts for identification. And I know in the family Faridae they often have these sort of ruffles associated with them, so I'll stick with that for now. There's a lot of invertebrates that live in association with sponges. I think we just passed a shrimp that was tucked away on the inside. There's a snail up on the top. Some of that brown material that you're seeing is just pelagic organisms that are getting caught on the sponge. Oh, there's a benthic tunicate. I can see one of its tentacle sheaths with one of its retractile tentacles extended into the water column there. And there's the other one just below it. We've seen several of these on some of the bamboo corals. It's the first one I've seen on a sponge. And then a hermit crab. Oh, great. Alan Collins is in the chat room. He's got some knowledge on the sponges. He agrees it looks like a Faridae, but then there's also in the family Eurydidae that have this form. It's a beautiful colony. We also see a crinoid on the lower surface. Now we're swinging around to this Metallogorgia. You can see the arms of the brittle star extending. And so, yeah, maybe this is a sort of a youngish colony because I'm seeing a lot of the branches on the upper left side look incomplete. And so maybe they're still just developing. And if we were to come back here in 5 years, 10 years, perhaps, maybe those lower branches would be dropped. I don't know. Nice view. Yeah, so what's interesting is that the lower branches are so thick, and that's what I'm kind of surprised at. That doesn't look like a branch that you could easily just drop off. Usually it's a much thinner branch. So I see an arm down there. If we could zoom in on the lower branches, I'm seeing an arm of a brittle star, and I just want to see, yeah, so there is a second one in there. How interesting. Exactly. Thank you for reminding everybody. It's very rare, and you know, all the time we say that there's one, but in order to reproduce, there has to be two at some point, or they have to be in close enough proximity to exchange gametes. So this is a really cool sighting. You can just see the arm of this larger brittle star in the upper colony, and there's its main body right in the center. The 5 arms connect there. The mouth is away from us. I think there's only one here. It's just its arms are so wrapped around. And another thing you see here, there's a lot of skeleton sticking off to the left with very few polyps, but there is a coating of tissue on them, so they're not dead. And so I'm just wondering if they're still developing. That's great. Pilot, we have a request to get a piece of that sponge. And I think it's really delicate, so I wonder if that's also a suction job. Thank you. I'm not sure that it's possible, Pilot, but Shore is saying, you know, if we could get one of those ctenophores, that'd be great. But I saw that on the back side, so I'm not sure that that's possible. That's fine. Yeah, I think that makes more sense instead of trying to get around there and breaking it all up. Les Watling came on the chat room, and he also says 2 heads and 2 brittle stars on that Metallogorgia. Crazy. Manuela Ramos also says exactly. Never seen a Metallogorgia like that. You know, you got that smoother. If you want to drop it in a box, I see there's a hard part there. I don't want to get the... Yeah, okay. It's just there's a piece of skeleton in it. Nice job, pilot. That was gorgeous. There they are in canister 3. Right, okay, we took 2 sediment samples. This is sample 4. The 2 canisters were both 3. Thank you. Okay, so let's get just a quick view of this coral that's just coming in below the lasers right now. Lasers are on it. I think that this is a black coral Leopathes, but we'll just verify that with a view. And sure enough, one of the things I look at is it's kind of this wiggly, thick main axis to the skeleton, and then very small side branches, tiny little polyps. And unlike a lot of black corals, this skeleton is actually quite smooth. That is, the spines are quite short. Remember, black corals are also called thorny corals. The spines are quite short, and they're prevalent on the small branches, but on that main axis, they've been, the number of layers have been put down for whatever reason. That axis becomes quite smooth. Thank you for that, pilot. And this is a really old sponge. I'm not 100% certain, maybe the sponge folks I'm sure can help me. I'm not 100% certain if that's 2 sponges, a yellow one growing on a white one, or if it's one large yellow one where the whitish brown is dead. So I'll have to get some input. And there's a crinoid growing on the top of it, we've seen this crinoid now several times. This is a stalk crinoid, so it's in echinoderm, related to the sea stars and the sea cucumbers and the brittle stars. And this particular type of crinoid grows on a stalk that it produces itself. And each time we've looked at it closely, there have been other organisms that are living on the stalk, basically fouling. And I can already see it's kind of fuzzy up towards the top. And once again, it's covered in this hydroids that I've been referring to as snail fur. I believe that's the family Hydractinidae. And for comparison, I believe there's another crinoid, and I can't tell if the crinoid is on the rock face or it's on the lower part of the stalk of the stalk crinoid. Now we're looking at the sponge. Yeah, so you can see all this, the brown stuff is the sediments that are moving through the water column, maybe being stirred up off of the bottom. There's mucus that the sponge produces, and so the combination of the mucus and the sediments and then all the gelatinous organisms in the water column get kind of caught up on the sponge. And if it can't wash it away, those cells will die. So here we have a nice interface between the white sponge and the yellow sponge. And the question is that two sponges competing for space, or is this one sponge that has two different color morphs? Okay, I'm looking around and I see some yellow stony corals. They're on the right. I believe that's Enelopsamia. I saw several of the Bramble Bamboo corals. We saw many of those yesterday during our dive. I think just on the lower part of the screen, probably the Acanthogorgia. We also saw that quite frequently yesterday. I'm not sure about this fan on the just below lasers on the left. That's probably, well, I'm not sure. Let's have a look at that. Oh, I do think I see an Acanthogorgia, though, right in the front center. And that was a target for collection. There we go. So this is another scleractinian, which means it's a stony coral. And the top of it is dead. There's some other colony that's growing out perpendicular to it. And then the color of this colony, I'm not exactly sure what it is. If it's a different species of Enelopsamia, it's quite possible. All the polyps are on one side of the skeleton, and it's a very flattened skeleton, planar. And so that's entirely possible. The yellow stuff looks like, I can't tell if it's hydrozoan or if it's bryozoan. I think it's bryozoan. So I think the yellow is a bryozoan colony that's growing off of some of the dead part of this colony. Rian Waller, who knows a lot more about scleractinians than I do, says that this is a sickly-looking Enelopsamia, where Enelopsamia is the genus name. And there's a very small crinoid there. This is the other type of crinoid, the feather stars, the comatulids. They don't have a stalk, but they feed in the same way, orienting those arms up in the water column to capture food particles. And then Pilot, before we came in on this colony, there was another one in the foreground, much smaller, wispier, right there. Bingo, that sort of brownish one. Thank you. I would agree with what Rian Waller just said. She doesn't think she's ever seen an Enelopsamia of that color before, and that's why I was so hesitant to give it a name. Yeah, so this is an Acanthogorgia, and you can see around the tops of these polyps, they look kind of spiny, and that's the characteristic crown of thorns. So this was a collection target the other day, and we also heard from James Aubrey that there may be 2 different species here, and that none had been collected at this depth in the central part of the Atlantic, I think is what he said. Maybe they were collected from shallower in the northeast Atlantic. So, pilot, if we can get a snip of this, thank you. This is another one I think that would lend itself well to a snip and slurp if you want to do it that way. Thank you. Oh, James is in the chat room now, and he said this actually may be a 3rd morph compared to what we've seen in our earlier dives. And Manuela's in here, and I think it was Manuela that you had said on, maybe it was dive 5, that Acanthogorgia was one of the things that you were looking for to ground truth for all the drop camera images that they get. So this will be a good collection. James is saying that it's a much smaller colony than I thought. James says he doesn't recall seeing any with yellow coloration in the tips of the tentacles. If that's easier for you, that's a real bonus for us because, sure, if it moves. It's a lot smaller than I thought when I first saw it, so. Just a rock. Yeah, let's make sure that it's on that piece you just dislodged and not behind it.
Now that rock is significantly
coral-like?
Yeah. And lighter and whiter and not covered with kind of the decomposition of organic material that we see around it. So I...
Understood.
I think
Great. Thank you.
I'm going to guess that rock's pretty hard. I'm also going to guess that it didn't come from here.
Yep, no question. Yeah, I can see what you're doing. You're trying not to crush it. Yeah, absolutely. I understand. That was beautiful, Sean. Nice work by Pilot there, and it testifies to all the hours that they spend working with this manipulator arm. I've seen them on the deck picking up little cups of water and tennis balls and various other bits and bobs. Great collection, and so we actually got multiple corals there, which will be really nice for future identification. As I said, the scientists at the University of the Azores are doing a variety of studies on the communities that live on these seamounts and island slopes, and very often the tools that they're using don't have the capability for picking up samples, and so we make our best guesses that we can, just the way Ashton are doing from what we see on the video, but having a sample on hand means a lot. It means you can put an actual name on it, and then you can go back to the video and more accurately quantify the biodiversity. Yeah, so we've seen this now on a couple of the dives, I guess 3 dives at least, I think, that have been in this depth range between 1,500 meters and 800 meters. And it's really interesting. We know it's in the family Plexauridae, so it's an Octa Coral. You can see that is a colony. It has several polyps. Each of those polyps is a set of tentacles, and there's a mouth in the center of those tentacles. So each of those is a feeding structure. Each of those is also capable of reproduction. The interesting thing is that it is unbranched, and we are not used to seeing plexaurid octocorals that are unbranched. I believe it was dive 1, not certain, but there was an earlier dive. We saw a plexaurid of this color that had a very long stalk, and then the branches came out near the tip. So my suspicion is that this is just a younger stage of that coral, and we did make a collection of that, and now we've been seeing them several times, and so we know that it's not uncommon. Very interesting set of tubes on the base there, probably built by a worm or possibly an amphipod, which is a small crustacean. It's a beautiful color, of course. Go ahead, Survey 1. So there'll be associates of the Acanthogorgia, and the one that I could see easily was Anthothelidae. Let's have a look at this tall colony, the bottlebrush, on the left side. So this is going to be a Chrysogorgia. I'm not sure that we've seen... Oh, all right. Cue the shark. We saw a cat shark yesterday. Can't tell if that's the same or if that's something larger. Has that same sort of really pointy snout.
He looks like a happy little shark.
Nice view of the lateral line cells there, the electroreceptor type cells. Thank you for that. We are getting some information. So there is a purple octocoral called Victorgorgia, which would not be a plexaurid. That would be in a completely different group of the octocorals. And apparently there are at least some unbranched plexaurids. So we'll have to learn more about that, get some more information. So a couple of the scientists on shore, Mike Vecchione, Michel Schauer, suggest that that is a lantern shark in the genus Etmoteris. And that is a deep-sea shark, so that would be great. That would be our first observation. So what we're looking at here is a Chrysogorgid Octa Coral, family Chrysogorgiidae, and we've seen many species in the genus Chrysogorgia. I think this may be the first observation of this particular species. Very dense branching. You can see the side branches are quite thick. It has a second main axis, and I remember seeing something like this in the New England Seamounts that I believe Les Watling and one of my former students, Eric Pont, described as a new species. That's a really nice lighting because you can see how the family got its name. The Chrysogorgiidae translates as the golden corals, and you can see the color of that skeleton shining from below the very thin layer of tissue. Also, like I've been saying now on several of these dives, notice the base of the polyps look like little saddlebags. You see these sacs that are hanging over each side of the branch, and that means that these polyps are packed with gonads. They're getting ready to fertilize eggs, and so that would either be packages of eggs or packages of sperm. I don't know if this colony is male or female. I do believe that in these Chrysogorgia they sort that way, meaning that the colony itself is not hermaphroditic. And then that also tells us that it's been there's been a lot of productivity in the overlying water, a lot of energy available for it in order to produce all of these gametes. Very nice, very nice, very healthy. And one of the things that would be interesting, Ashton, if we could come back here several times during the year, would be could we see some change in that reproductive status? And all you'd need is an image like this. I mean, ideally, if you did the histology, you could say more about what kind of gametes there are. But the question would be, is there a seasonal pattern to that reproduction? Mm-hmm. This is the midsummer. We've had our spring bloom in the overlying water column where there's lots of production of phytoplankton. And then maybe if we came back in the fall or winter, we'd see that they're not producing eggs and sperm anymore. Or maybe they are. And that's the question. Are they seasonal or are they continuous? And early on, people thought that in the deep sea they would be continuous brooders because you don't get a seasonal signal. They're outside of the lighted zone, so how would they know? Well, now with further studies and using things like sediment traps deployed in the water column, we know that there's a pulse of food that descends from the shallow water, and so that in itself is a seasonal cycle.
Yeah, so there's
seasonal signal, excuse me.
Yeah, so there's a seasonality to the overlying primary productivity, and that makes its way down into the deeper ocean.
Rian Waller says, if only we could tell seasonality from images, my life would be so much easier. And it's so true. I mean, she has just now, I believe, flown from Norway down to Chile in order to look at corals that are undergoing reproduction. And if I'm not mistaken, she said she made a stop in Alaska along the way to do some other research. So it's not easy getting yourself into a position where you can study these corals. She's gone down to Chile because they have these deep fjords there where the cold water comes up really close to the surface. So it's an analogous environment to the deep sea, but it's shallow enough that you can dive and make some of these observations and get the right collections. Looks like we have a grenadier fish in the distance. Yeah, and I just want to specify to everybody that I'm offering hypotheses and making predictions. I'm not saying that there's seasonality here. I'm saying that if we had the tools, we should come back more often to find out, is there seasonality? Or is it just a continuous reproduction? Or is it just, maybe it's aperiodic. It's when we can get enough food to make the gametes, we will. Maybe we can have a look lower left. There's some kind of feathery-like, kind of sad-looking colony. It looks like a Bathypathes from a distance, but also very pale in color. I'm pretty sure that's the same grenadier that we have been imaging on several dives now. It's got that really pointed snout. I think it was a Coryphaenoides. Yeah, the one on the left. So it's actually an Iridogorgia it does not look healthy. The color is just not right, and I'm wondering if there's not even any polyps left, or I can see actually a few.
What's coming, video?
This is an Okeanos that's on the way out,
okay.
so it's mostly dead. You can see the skeleton is there, and it's kind of shining. It's sort of gold color. Belongs to the same family, Chrysogorgiidae, as our Chrysogorgia we just imaged, but it's mostly now covered in this material and sea snow that's drifting through the water column. And there's only a couple of side branches that actually have live polyps on them. So there you get a good idea of what the skeleton looks like when the tissue is removed. There's one amphipod there. And if you pan upwards, Pilot, you will see that there are a couple of branches
Can we go up, video?
that have living polyps on them.
Copy.
There we go. There's some right in the foreground. So 1, 2, 3, 4, 5. I
Copy.
only see 6 there. And then notice these do not have those large saddlebags on the side, so these don't seem to be undergoing reproduction, and that's not a surprise. They're probably just trying to hang on and survive. And if we go a little bit higher up and on the right side, I think you'll see a couple of other branches with some polyps.
Copy. Going up.
Yep, there's some more. Yeah,
Right
so
there.
just a scattering. So, you know, this may be a senescence thing. We don't know how long these corals are able to live. And then there's a nice stock crinoid on the left side that's using the coral as a perch.
How long
It's
ago
not
did
a
that
stock,
ship
excuse me,
move in?
a feather star, an unstocked crinoid, and actually using.
6 minutes ago. Okay.
Oh, I see. There's a bunch of barnacles. I thought they were on the crinoid, but they're actually on the Okeanos. All right, that's good, Pilot. Thank you.
All right, I will clear video. Copy. All right. Lifting off. And then I'm going to come back.
I think I'm seeing a couple of primnoid sea fans also up ahead. Those are the relatively large white ones that you see on the left side of the screen.
What do you see geologically, Scott?
I see a lot of what I would call erosion and broken up features. And I'm seeing a lot of coral rubble that is blackened, and I don't know if these frilly features, I'm pretty sure they're not, but I would, just to give you an opportunity, could it be a broken open pillow, for example?
Yeah, I don't think we see any broken open pillows. It's everything's so weathered and kind of post-depositionally altered. It's really hard to tell. But I really see kind of 3 main geological features, and that's the really fine-grained white calcareous sediment that we got a really good close-up view when we did the suction of the holes. We see the kind of medium gray, darker gray coral rubble, as you mentioned, and then we see this much more darker black material, which I think is just the partially diagenous concretion of that sediment that we saw earlier. It's very similar to what we brought up in our previous dive. I think if we looked at it, we would see a bunch of micro-scale calcium carbonate that's been precipitated, remineralized, probably post-altered.
Can we zoom on the fish on the left side of lasers tucked in just behind the rock?
And I think by, that close-up that we got of that sediment earlier, helps me understand what those larger scale blacker features are. We didn't really see when we were looking at that fine-grained sediment much fossil material. We saw some pteropod shells and things like that, but everything looked kind of micro scale, and so that's what I think is actually forming those carbonates. Turning off lasers.
So you're just getting so packed together and cemented and so on.
Packed together, but then also you have benthic foragers that are kind of, filtering them out, right? So that's a filtering process. You have precipitation and remineralization, dissolution of the minerals in them that is helping concrete them together. Then depending on how buried they get, you also have compaction, and that can be overturned.
I was going to say this might be Lepidion. We've seen that several times, but this one looks a little bit, at least grumpier to me, more wrinkly, so I don't know for certain that it's the same species that we've been seeing earlier. Yeah, Mike Vecchione says a cuskeel.
I wouldn't say grumpy. I would say slightly aloof.
Well, for sure. It's got that whole attitude going.
Yeah. but we kind of just walked in on his morning walk.
Yep. Looks like his head is just over a yellow Hartwigia sponge.
Let's come on video.
Looks like he's got a bit of an injury down the side of the body, and many of these fish have parasites associated with them, although that is not something that I've been seeing here on the Mid-Atlantic Ridge so far. Don't believe I've seen any fish with parasites. Thank you, pilot. That was good. I did think I saw, noting this for our shoreside folks, I did think I may have seen a hemicorallium or corallium as we were passing over, so I'll make sure I look out for that going forward. Looks like an Acanella down there, bamboo coral, some more Anelopsamia. So I should note, we landed basically on a small ridge on a bigger, what would you call this, a mound, topographic high, part of it. Sorry, I'm looking at the 3D perspective. So if you look at the overall dive track, we're on kind of a cone, and we're on a small side ridge on that cone. And when we landed in a relatively flat spot, and as you can see, the slope is increasing, and probably 100 meters or so upslope, it's going to get even more steep. And so it'll be interesting to see if there's a transition in the community. So let's get a look at this upright stick, this whip right in the center, please. Pilot, not sure if you heard me, a view of the stick on the, now on the left.
Sorry.
No
this
problem.
Tall one right here.
Yes, thank you.
Sorry, Scott. I hadn't heard you.
No problem.
Okay. Approaching it video.
This is almost assuredly a bamboo coral, unbranched, or at least so far unbranched. I've talked about this several times during this expedition. I think there's also a bramble in the foreground. We'll look at that next. But the question, of course, is these are all snapshots in time. Okay. And so it's possible this could begin branching later in life. They live for a very long period of time.
And turning off lasers.
Our best understanding is that there are some species that never branch.
Okay.
And the difficulty is sorting out what are we seeing, young versions of ones that haven't branched or actual unbranched ones. And so you have to look at other characteristics besides just the gross level of there's a unbranched bamboo coral. You need to look at some of the details of the polyps. So they're at least very dense, these polyps. I can see some very prominent needle-like spines between the tentacles, and the tissue itself is relatively opaque. It's hard to see through, so that's a good characteristic. In fact, as we go up here, I can't see any of the nodes because that tissue is so thick. So that's good. Thank you.
I lost my footing.
And then just in the foreground, lower on the rock, there's some very wispy, it looks like a tangle of very thin branches.
To the left?
Nope, just keep going straight down. It's right on the bottom of the screen there now. Yep, bingo.
Go ahead, video.
So I think this is another kind of bamboo coral, one we call the Bramble Bamboo, although that's just a sort of a descriptive name and covers several different species. But we'll just get a verification of that.
It's like right between my toes. So
yeah, it's pretty small. I can already see that it appears to be branching at the nodes. Yeah, if you can tuck down any lower on it, that would be valuable.
Okay,
polyps are retracted, so that's actually pretty good. I'm seeing that it looks like two of these sclerite needles are longer than the others. Trying to get that zoom at the base less. We're not quite in the right position. And it's a pretty small colony. And this is another thing we have a question about. Do the bramble colonies stay a bramble when they get really big? Or do they take on some other morphology? And they probably take on some other morphology because we've never seen a giant bramble. And so I wonder if some number of branches become the dominant growth axis.
Okay, go in video.
I suppose another alternative is they never grow large. They always stay in these small little tangles.
And then where exactly do you want to zoom in?
To the right, and just sort of go into the mass. So not the tips. Yeah, keep going like that. Maybe a little bit higher to the right. Okay, that's good actually right there. So let's go in a little bit more. Just go a little bit right and stop there. Don't zoom anymore. A little bit right. Almost there. Not quite. More right.
Oh, sorry.
No problem.
Right.
There we go. Yeah, it's that bright white one that I'm looking at. That's perfect. So that is definitely branching from the nodes, really thin. You can see that different branches appear to be anastomosing, which means when branches come back into contact with each other, they kind of merge, the tissue merges, and so you get this weird branching structure. And I'll just point out while we're here that the white thing on the far right is a bryozoan colony. And I'm pointing that out. We're done here, pilot. Thank you. When the video's happy. I'm pointing that out because the bryozoa is a completely different phylum from the Cnidaria, and yet it has very same growth morphology. And all these organisms are responding to the fact that they are getting their food out of the water column, and there are some, how would I put this? There are some morphologies that are well adapted to getting up off the bottom and getting a current flow and being able to capture that flow. And one is to create a fan, so you can spread out those feeding structures. You create a baffle, so as the water passes through it, it sort of spins back on itself and slows the current down so you can grab the food. And then having some tentacular-like structures that you can reach out and grab. So that's one way of doing it. Other organisms may put out a mucus net. And then there are things like fish and whales that swim through the water column, open their mouth, and basically create that filtration system inside their mouth. What's going on in that rock up there, Ashton? It's got a white edge.
It does have a white edge. You don't think that's biology?
I thought I'd check with biology or geology first. I was hoping it was geology.
I think it looks like it's covered in little frilly sponges.
It actually looks, I mean, I'm sure it's something tipped over, but it looks like a massive, giant sponge. I wonder if it is, and that's the back side of it. I was sort of joking around that I was thinking the sponge was overgrowing on a rock, but I think that whole thing is sponge. Do we have the lasers on?
Wow, look at that.
There we go. Oh, that thing is huge. 10, 20, 30, 40, 50. It's more than a meter tall and a meter across. So you're right.
I know.
It's biology.
It's biology.
I know I was. I love that.
I can't always tell what biology it is or what rock it is, but I can usually tell whether it's rock or not a rock.
This may be... No, it's probably not the same as the one that we sampled earlier today. I don't think.
Full specimen right here.
I don't know if it's the same. I don't even think it's the same as the amphitheater one that we sampled earlier.
Video of the.
And is that secondary organisms encrusting the inside of it?
Yes, it is. There's lots of stuff overgrowing, from other kinds of sponges to octa corals to stony corals. There's maybe even a large snail in the center. There's several echinoderms, both crinoids and ophiuroids.
Is the snail that spheroidal?
Yeah, the spheroidal kind of brown gold color.
I'm gonna try to do it.
Ian Waller says this sounds like a game show. Rock or not a rock?
Rock or not a rock?
Ready for some fog lights, pilot?
Yeah.
Fog lights coming on.
Even an Acanthogorgia up at the top.
And it's amazing just how delicate those exterior structures are. I mean, just slightly.
You would have thought that this was rocky as we're going. And look at the wave.
You described something the other day as sheet-like, and that's kind of what it reminds me of.
The sponge?
Yeah.
Okay, sorry, video. No, I'm gonna try to like
Habitat
hover
farming
right here.
sponge for sure. This is one of the comments
Go
from one
ahead.
of our scientists on shore.
We
I
can
completely
zoom in on
agree.
this part
We got
right
hermit
here.
crab also. I'm kind of interested in that globular brown thing in the center. Now I think it's an anemone. Oh no, it's a sponge. It's another kind of sponge. How about that? Here's some dead cup corals in here. I mean, this has been growing for a long time, so it's probably a mixture of stony coral and sponge together. So I don't think the whole thing is a sponge. You can see how diaphanous it was. I don't think it would support all this other stuff. Yeah. You can see that all this branching. So I'm not sure if this is a very old Selenosmilla or probably that more likely that than it is Ophelia. But so the underlying scaffold is an old dead stony coral, and then you've got this massive sponge on top of it, and then you've got all these other organisms growing on it: crinoids, ophiuroids, the brittle stars, octa corals, hermit crabs. Just naming the things as they're passing by. Rian Waller notes that, you know, there are several cup corals in there as well. So we have solitary corals on top of the colonial coral. Here are some, there's an Acanthogorgia on the left. What's going on this crinoid? It looks like there's a couple of snails maybe feeding on it. That's interesting. Huh. Don't recognize the snails, gastropods. What else can we see on here? Impressive feature. You know, I'm not sure how fast or slow these sponges grow. I suspect it's pretty slow, so I would suspect it's pretty old, but don't know. That thing in the center, that brown ball in the center, that's another sponge. You can see that it's got a little siphon at one end. Oh, the lower, now it's in the center, but below the lasers, there's a very smooth kind of gray-blue object. I believe that's an Acesta file clam. And I was hoping we'd see more of those. We had a request even before the expedition began that if we saw any on the Mid-Atlantic Ridge, it'd be really useful to collect because they believe there's a different species here, but they haven't been able to get a sample. Yeah, tucked in right underneath there. It's possible it's a brachiopod. Now that I'm looking at it, there's something about it that's saying brachiopod to me. But it's closed up, so we can't see if it's got a lophophore, which would be the tentaculate feeding structure in there, or if it's got gills. That's cool. All kinds of stuff tucked in there. Boy, wouldn't, you know, we'd be working till next week processing this sample. There's a Galatheid crab underneath that bivalve. Different kinds of sponges. Now I'm even seeing carnivorous sponges tucked in there. There's a stoloniferous octocoral. That's the one that grows kind of like a ribbon instead of having a skeleton. I mean, it's... Oh, and I think one of those large solitary tunicates just poking its siphon up,
I'm gonna start lifting up
behind on the left. So a really nice view of what we call biogenic habitat. And that means habitat that is created by animals that are growing. Actually, it's any organism, but down here in the deep sea it would be animals, but on land it would also be plants that are creating a substrate for other organisms. Spectacular. Oh, perfect. So Jean-Marc Gagnon has joined us in the chat room, and he's at the Canadian Museum of Nature. He's a curator there, and he was the one who alerted us to this file clams, the Acesta, and he has seen this himself, and indeed, that was the giant file clam that they're really interested in. I'm afraid, Jean-Marc, that is way tucked in underneath there. We're not going to be able to get that one. But stand by, Jean-Marc. Yeah, go ahead, pilot. Go ahead, pilot.
I turned off OKEX, so
copy that.
I think I might not be listening to you. So,
Yep.
would you
No
talk to me
problem.
directly?
I will do that for sure. And so that was a really nice view of that sponge. And I was just talking with a colleague on shore who noted that that's the giant file clam that they're looking for. That thing was tucked in way underneath that feature. We couldn't get into it. But now that I have the
there you go.
knowledge of what it is, I will keep a good eye out and hope find another one. So if you are going that direction a little bit to the right, hanging off the side of this next topographic high, let's have a look at the fans there, the sponges there.
Copy.
Thank you.
I think you're muted, but yes, that's good.
Yes, I was muted. Nav, can we get another 10 meters, please?
We can. 050 again.
Yeah. Sounds pretty good.
Yep, sounds good. We'll do.
Actually,
So this is this
40,
rock's interesting. We
please.
have
Yeah,
two
more north
large
towards waypoint 10.
fans,
Yeah,
and
40, please, thanks.
I kind of feel like that top fan is a bamboo coral. Maybe both of them are, but you see all the purple.
That's a beautiful.
Yeah, it's just a gorgeous color, and there's something about purple in the deep sea that everybody gets this reaction to it that it really stands out.
Approach it slowly, video.
And I'm also seeing another Acanthogorgia colony, I believe. We just collected one earlier. That's the sort of
Lights
yellowish
coming
brown on the lower side of this feature. And then the bulbous
On
white
halfway.
stuff you're seeing are all sponges.
All right, let's go up, tilt up, and then let's go right here for the. Actually, hold on 1 second. Let me just get settled.
So the way this purple stuff is growing, it sure looks to me like it's mat-like and it's overgrowing. The whole structure that you're seeing, again, I think is biogenic. So it looks to me like it's either an uplifted
Copy that. Yeah, let me
chunk
know.
of coral
Pilot, I would just say
or
watch
coral
your
that grew on some
carbon
unlifted
may
piece of carbonate.
impact that
But the
coral.
base is coral. And so the purple that you're seeing is overgrowing kind of like a mat. And so that's telling me that it's probably Clavularia or
Yeah, I'm having
Trachythela
so much,
and not
such a hard time. Just,
Victorgorgia, but I'm not
loading.
100% certain about that.
so I just keep on backing. Yeah, let's do partial then.
It is an Octa Coral.
Okay.
I'm also seeing some more of the cup corals. There's
Yeah,
actually a
maybe
little
we
bit of the
won't be
stony
able to
coral
go
that's
like-
alive there. I don't recognize the sponge at all. The texture, especially that upper one, with all those sort of ridges and lines in it. And hopefully there's some sponge biologists on the line that can
Yeah,
help me
let's-
with that. I think the stuff that's waving at the top of this feature
Try to
is
stop
probably
going up and down.
colonial
Okay,
hydroids.
let's try this. All right. Let's come in on the sponge. Yeah. Copy. Oh. Okay. Tilt down.
And once again, we're getting a picture of just how much diversity there is when you look at some of these surfaces. And you get a close-up look at the number of different species that are associating with these habitat-forming species. And so here we have sponge that's growing on top of coral that's serving as habitat for brittle stars and hermit crabs and worms and
All right.
this.
Then let's try the purple coral to the right. All right, let's come in slowly.
I think the cup coral that's tucked in there beside that white sponge, that one might be alive. See some orange color.
Too much.
We're getting a look actually at the bamboo coral now.
Yeah, it's.
Actually,
I
first
think
we'll
I'll
look at
drift
this
it back,
purple.
so. Okay, trying to hold
Yeah,
there.
what do you folks think on shore? Is this a Victigorgia, or is this Trachythela? I can't tell if it's got its own skeleton. So Victigorgia does produce its own skeleton. This one, maybe it's doing so.
Right there, that's good right there.
It's a little hard to tell if it's just overgrowing something else or not. So here's the bamboo coral. This is almost assuredly in what we call the J3 clade,
Going there.
Jasonisis, or Dokidisis. It's so old that the nodes have been largely overgrown, and I could see them down low, some thin lines that showed that the calcium carbonate internodes above and below the nodes had basically obscured them. And the other thing is that the tissue is filled with these very small scale-like sclerites that reflect the light, so we can't really see through the tissue. It makes it less transparent, makes it more opaque.
Go ahead.
Not seeing too much in the way of associates on the bamboo coral.
Is there anything else you want to take a look at? Watch lead.
No, I'm satisfied here, thank you.
Copy. And then right, I'm moving back. Sure.
Jean-Marc Gagnon says, incredible assemblage of so many species.
Right.
Absolutely right. And note that they were raised on this topographic high point, and now as we look back down at the low point, which is quite flat,
I'm having a lot of trouble just
there's
like
many fewer species, or at least species that we can see.
Like
that is, there are fewer of the macrofauna or the suspension-feeding macrofauna.
Because I feel like if I stay like this,
I think we're seeing more lines of holes here. Is that what I see down here?
What are you talking about?
Pilot, lower right corner. Look in the sediments. Right. The right corner. Starboard.
What's going on today with me? Okay, sorry.
No problem. Yeah, so I think there's another line of those holes maybe. Can you put the lasers back on?
Right in front of us?
Yeah, I need to see the laser. Yeah,
this
there.
One right there?
Yep, exactly.
Okay.
Oh, there's another one over here. This one's more curved. I think that's what they are. We'll see as we get over them if they've got the square holes or not. I think we might need to come up a little bit higher. Don't usually say that, but yeah. I mean, so those ones I think are so old that they've completely
infilled. You can zoom in on mini zoos. It's like right there in the middle of the screen.
Yeah, it's like a trail, it's like a path, but I don't see the distinct holes. Okay, can move on. Thank you.
Okay. All right,
so just before that, I was starting to say that on this flat area, you don't see the large megafauna suspension feeders. That doesn't mean that there isn't a lot of diversity here. It just means that most of those organisms are in the bottom, and they're probably much smaller. And so we would need different tools in order to see them. Every once in a while, just like right here in the lower right and in front of that, you do see some sea pens. So these are octa corals that are adapted to live in the soft sediment. There's a few species like that. That's the genus
Sorry,
Penatula.
co-pilot, you're
I
muted.
see at least two of them.
Yeah, like I cannot hear you, Sean, at all. Like I don't see you muted, but I can't hear you. Nothing.
Nothing.
Try to talk on RV. I'm listening to everything, but go ahead. Nothing. Right there. Well, I see an open mic, but
Test, test.
there you go.
Okay. Yeah, you want to do 20 meters, 040, pilot?
Yeah, that sounds good.
Sounds good.
And then,
Copy.
do you want to zoom in right here? Watch lead.
No, we can go on. I know what this one is. Penatula.
Copy.
Thank you. Well, if these aren't the same kind of lines, they're certainly at least individual pockmarks that are moving, so that's kind of interesting. And the shapes are now more varied from what we were seeing before. But we're still not, I haven't seen any single hole that I would call rectangular, but otherwise, sure looks like a similar feature. Yep. Yeah, those holes. Yeah, look. That. Yeah, that looks like it's the same thing right there. This one will be interesting.
All right, pilot, you're nearing the end of your leash.
Copy.
It's.
All right.
Ship move was in about 2 minutes ago, so hopefully we can start feeling that soon.
Copy. I'm gonna raise a little bit watch lead, and we can watch it from here. As I get a little bit more tether, I'll get close.
Okay.
Mini Zeus is right on it, so you can look at it from a higher perspective.
Thank you. Yeah, this one's a much more complex pattern.
Go ahead.
So here in the control room, we're looking at a second camera that's mounted higher in the sub and has a steeper angle to the substrate.
So I'm gonna rotate.
And again, these holes are largely infilled, but they otherwise fit the pattern. If we could just ask the sea pen that's sitting next to it what made them, that would help.
Tilt down on Mini Zeus. Copy. Hold on 1 second. Okay, so this is where we were, tilting down.
Yeah, part of the problem here is that, you know, Aston said this earlier, we don't know how old these particular holes that we're looking at are. And so it's entirely possible we see some of them have been infilled with material, and so any organism can now be using it as a burrow. So we're just kind of, oh, actually, pilot, can you look, since we're still hanging out here,
Yep.
at about 7 o'clock, there's something sticking up from the sediments.
On the main camera.
On the main camera, correct.
7 o'clock.
Yep.
I don't see anything.
It's about halfway. You're getting close to it with the lasers. And
underneath the lasers?
now, yep, between the lasers.
Copy.
Thank you.
Let's zoom in. Yep.
It's a black coral, I think. Yep, that is a black coral whip, and it's probably attached to a small protruding rock or a cobble, because this particular species I don't believe grows in the sediment directly. Thank you for that.
Moving back and forth. Copy. All right. So, do I have a little bit more tether, co-pilot?
A little bit.
Copy. So, from here, watch lead. Do you want to take a closer look at any holes on the main camera, or do you want us to...
They all look pretty weathered to me.
Copy.
They don't look fresh enough to spend too much time on, but it's good that we have them on the camera.
Okay.
Last move was just complete a minute ago. Would you like another one?
Let's hold on a couple minutes maybe.
Copy.
Although there's nothing up there, so do you want to keep going?
I don't see anything. Up to you. It looks pretty barren. So
All right.
Could probably keep moving safely.
Copy.
Another 20 meters, 15 meters. What would you like?
Another 20 at 040.
All right, I'll do it.
Okay, tilting up. I'm going to my starboard side.
So just as a reminder, we're kind of moving up slope on a small ridge on the side of this much larger cone-shaped feature that we're climbing today up to the top side about, what, 1,400 meters or so, is that right?
1340.
1340. I don't know that we'll get to the top, but on the way, the slope that we're climbing will or should get significantly steeper.
ROV, could you give me a range to waypoint 1, please?
Well, actually, do you waypoint 1 or waypoint 2?
We haven't reached waypoint 1, correct?
Or is that the launch point?
We'd landed at waypoint 1.
Okay, yeah, I guess we do it differently in our dive summaries. Yeah, waypoint 2.
Yeah, we're about 40 meters away.
Okay, thank you. So, Scott, do you want to dogleg it after waypoint 2 to waypoint 3, or do you want to continue upslope?
I think I'd like to dogleg it over to that higher slope.
Okay, so we'll continue the dive track as planned. Okay.
Okay, almost to north.
Yeah, first slope series is picking up is about 50 meters out. Cut out now. Nothing too serious looks like ahead.
And if RV, if you have the opportunity, if you could just do an occasional close-up on the sediment whenever you can, I'd just kind of like to get an idea of what actually makes up that surface.
Copy. Let's hold on 1 second, video. All right, let's come in. Quite a bit.
Yeah, so it looks, and look, there's the one purple Octa Coral. Yeah, it looks much less finer grained than what we were seeing earlier. Maybe a larger percentage of pteropod shells or larger scale concretions, and you can imagine all of this material kind of consolidating together and through biological and chemical processes, consolidating into kind of that black crust we see in other places. Hey, look what I found you.
We can come up.
I don't know what it was, but I'm pointing at something biologic, and I wanted to get a sense of what the actual the size of the detritus that we're seeing now, because it looks to be significantly less finer grained than what we saw on the lower slope.
Are they pteropod shells and things?
They look like them to me, but I'm no expert. When we start getting into these sedimentary or we won't find them here, but metamorphic processes, that's where my knowledge base really starts to get stretched out.
Sure.
Yeah.
Looks like is this possibly an Bramble bamboo coral popping up here in the center of the screen. It's got that characteristic brownish gold polyps with the very bright white holdfast.
Let's partial zoom video.
Yeah, that's absolutely what that is. It's a bamboo coral. Looks like it has a shrimp in its branches. It's really common. Acanella, that's probably Acanella arbuscula, is one of the easier corals to be able to identify from a distance and in video. Metallogorgia is another one. Iridogorgia is another one. They have enough regularity in their shape and distinct enough from other corals that you can tell them apart, even though there is some flexibility in, you know, the details of how they branch. Oh, what's that wiggly little thing?
Comment right here on this dark subject video. What is this?
I guess it's a... So the really dark things in the center are dead cup coral, nice shrimp, thread-legged shrimp.
Then there's a tiny little coral at the bottom.
Yeah, so I'm not sure. It's an Octa Coral, but I'm not sure which one it is.
Okay.
So small, and all the polyps are retracted. Looks, yeah, just not sure. There's a hermit crab. And then they all must be on just a larger piece of carbonate, I guess.
Can come out a little bit video.
Yeah, it's interesting to see that really small coral. So one thing to think is that, or remember, I should say, is that they all have to start as larvae. So they're starting as, you know, a small ball of cells that settles on the bottom and then begins to grow.
Center up on this
and we often miss those really small ones just because of the mode of operation. You know, we're not doing microscopic views at the bottom all the time.
Real slight.
Yep.
But when we do see these really small ones, it's quite satisfying because it tells us that there's some new recruitment going on in the area. Now that didn't look like too stable a substrate, so it'll be interesting to find out how well it can survive, you know, over a decadal scale. We'll be able to grow there.
Partial zoom on this black subject video.
I'm going to guess this is a sea-pen peninsula. Let's see what happens when we zoom in on it a little tighter.
Okay, I'm still approaching it.
Huh?
Tilting up.
That looks like a sea cucumber that is standing up and folded over. What in the world am I looking at? Oh, it is a peninsula that is bent in half. Oh, how odd. So this is a sea pen for sure, but it's folded on itself, and all the lobes are hanging down. I mean, to me, that looks like maybe what a peninsula would look like if it fell asleep on its feet or was dying. Yeah. Oh, that's really weird. So yeah, there's some on shore. People are also thinking that maybe it's dead, but that the tissue, it hasn't been dead so long that the tissue is, you know, disintegrating, being decomposed. I can still see the individual polyps and so on. It's kind of odd. So it's odd because these peninsula have a single rod skeleton on the inside of the what's called the primary polyp, and it goes all the way to the top. So it's almost like it's snapped. So I wonder what would have happened to that. Huh. You see something new every day.
All right, video. I think we're good. pushing out.
It looks like there's something large standing up on the rock in the upper right. Yeah. Can't tell if that's a sponge or a coral. Yep. Sorry, I was muted, pilot. The big one on the right looks new to me. Yeah, this is the correct right this time. I see an Okeanos colony behind this. Oh, this is a sponge. So this is a large kind of, I describe it as a radar dish sponge. So it's still a little ways out in front of us, and just a reminder of the operations. So in order for us to make big moves over the seafloor, we have to have the ship do a move, which will pull Sirius, and then that will allow ROV-D2 to move. As we're waiting here a moment, and I'm describing the operations, it's probably better for the GFOE team to introduce themselves and describe the operations. So why don't I turn it over to the front row?
Sitting co-pilot currently is Sean Kennison.
Sitting navigator is Jonathan Allen.
Currently sitting pilot, Fernando Aragon.
Broadcast control is Brian Doros.
And videographer in the clipping chair is Anna Sagatov.
That's our team currently in the control room. Getting you these beautiful images. This pretty large sponge. You can just see these two red dots appearing on the sponge, on the back of the sponge. Those are 10 centimeters apart.
This sponge and the sponge we just saw earlier, those are the largest sponges I think we've seen on this.
I think you might be right.
There you go.
Yeah, which is interesting because we received from a reporter a list of questions, and one of them was, "What's the largest thing you've seen down here?" And I think the largest thing that I've seen in the deep sea is a sponge, about the size of the old Volkswagen minivans.
The largest thing I have seen in the deep sea is a volcano, or the seafloor. But it's interesting because we've dove in this depth range before on this leg, but we did not see the size of these sponges.
Yeah, I agree. Did we get up to 1480?
I think so.
Yeah, we did?
Yeah.
Okay. I can't tell. This sponge to me looks to be dead. There's too much sediment on it. I'm seeing a variety of these stick-like things. I believe they're either fan worms or hydroids. Let's see. Look like fan worms. So those would be little polychaete worms that are also suspension feeders. You can see, if you look closely, you see some of them. There's these little feathery bristles extending from the tubes. Those would be modifications of the head that allow them to capture food particles. But otherwise, the worms build those little tubes.
Yeah, so yesterday we were in about 1240 to 1090. And then I think
we had another dive. We were at around 18 to 17 something.
Yeah, 2 dives ago we were 1760 to 1640. So this is kind of right in between those 2 zones.
Looks like maybe a 5-arm crinoid, the purple one on the rock, and then a beautiful Ovidigorgia Octa Coral in the distance there. So this is commonly referred to as the feather stars, the Cachalote crinoids, a kind of echinoderm related to the sea stars. And unlike the sea stars, they move around with their mouth facing away from the seafloor. And they do have tube feet. The tube feet are really small, and they're on those little side branchlets. Those are called pinnules that are coming off the main arms. And that's what gives it that sort of fuzzy appearance, and you can see it now. Yeah, you see the pinnules extending, you see the tube feet extending from these pinnules. Tube feet are, when you look at them microscopically, are in these little sets of 3, and they will flick particles down into a groove that runs along that arm. You can see the grooves actually quite nicely in the arm, and they lead to the mouth, which is in the center of those 5 arms. Pentametricrinus is the name that I'm getting. I think we just passed over a baby Metallogorgia. There's another Ovidigorgia we're just going over now. A couple of sponges. This might be the Pharonema sponge in the center. Can we have a look? Oh, no, that's a rock. It's not just a rock.
Oh, no, it's a rock.
Actually, I think it's a can.
Yeah.
Just off screen to the left, looked like a rusting can.
Yeah, that color of oxidation you do see on weathered basalt glass, but that's a little too.
Yeah.
Now, if it's a bottle, it's always interesting. Oh yeah,
that's a bottle. It's got a hermit crab inside of it.
It's full of samples.
Yeah.
You don't want to collect that biology sample?
No, I'd like to pick up all the trash, but we don't have the space. I'm not sure where you're looking. Oh, there? Yeah, maybe. Coming into center right of the lasers all the way towards the edge of the screen. There's something very small and fan-like. Now it's below the lasers. Yep. And that looks to me like a Swiftia Octa Coral. So that's the first one of those that I've seen today. Remember the Plexauridae? You can see the polyps extending from the little humps. And Pilot, if you could just back up just a bit, zoom out, and then look below your skids. There's something very dark brown, smooth. There it is, right at the bottom of the screen. Yep, correct. I guess it's a sea cucumber. Not too many things with that texture. Oh no, it's a nudibranch. Look at that. That is not a sea cucumber. It's got 2 pairs of cephalic tentacles. So this is our first nudibranch. So this is a mollusk. Basically, it's a snail without a shell. Very nice. You know, I'm sorry, it might not be a nudibranch. It might be, I hope James is in the room. He could remind me. It's definitely a gastropod. And I'm trying to remember the name of the group that has the reduced internal shell. Devil-looking nudibranch, says Tina Sacco. Definitely a gastropod. I can be safe and say that. And so head end is away from us. Not all of them, but some of them. The nudibranchs. And that's why I don't think this is actually a nudibranch. A pistibranch, thank you. That's the name I was looking for. But more specifically, I have to go to my notes on this group. And there's an interesting urchin that it's approaching. So again, I said this before, every time we look in at the seafloor, there's all kinds of cool stuff that we see. Lots of diversity. There's a sea spider that the laser's just crossed over. Close your eyes, Ashton.
Mm-hmm. Now that structure above us, that looks like it might be an old weather pillow.
A couple of them.
Yeah.
So instead, we'll look at the sea spider.
Mm-hmm. Now it's interesting because we've been on the Mid-Atlantic Ridge for the last 2 weeks, and we've been diving in an area with pretty well-defined geologic and tectonic processes, so we can look at estimates of the age of the seafloor we're on by looking at the distance from the ridge axis. But now we're starting to get into this area where we have the confluence of 2 processes going on. We have Mid-Atlantic Ridge volcanism, but we also have hotspot volcanism. So, the island of Flores, which is about 50 kilometers east of us, it sits on Mid-Atlantic Ridge oceanic crust that's about 9 to 10 million years old. But the volcanism that formed the island is actually only about 2 million years old at the oldest. So, imagine where we're at. There's kind of a confluence of processes, and it's really it's not clear the age of the products that we're on. So that has implications on the amount of sedimentation that you might expect in the area, the likelihood that you might see these pillared outcrops. And I'm trying to say this, but it's hard for me to concentrate while there's this nightmarish-like creature crawling towards me.
I'm going to guess that this one's a male, and I'm saying that because you see those 2 orange kind of limb-like structures that are going backwards. They're really quite long. Those are what we call the Ovigirous Legs. And
do I want to know what that means?
I think so. It means that they hold eggs to create more babies and more spiders.
Okay.
Yeah, I thought you would want to know that.
Now, are legs the correct terminology?
Sure.
Okay.
They're segmented legs.
Okay. Are there segmented legs? Are those opaque, or are they? We saw legs before that were full of
extensions of the digestive tract. Digestive cechi is what we would call it. Yep.
So
it's a good question, right? I was thinking the same thing, that the ones we saw before were much more orange. This is much more white. And so my initial impression is that they are opaque, so
Yeah.
we're not seeing extensions of the digestive tract in there. It's passing over a little anthomastus. Do you want to have a look at those pillows?
Yeah, sure. We can. Thank you. Yeah, so that looks like an old pillow deposit. You can see it kind of flowing from that upward section. Yeah, let's zoom in on a piece of the crust and see if we can actually see anything texturally interesting. I imagine a lot of it's going to be coated in manganese given the age. And so, one of the things you're looking for either the sandy texture, which tends to be indicative of the manganese deposition, or you look for the glassy outer rind surface that we saw earlier. You can also look for little holes, little vesicles, which are a product of outgassing when the lava is in place. And you usually see those more on exposed surfaces. And maybe we'll see some more pillows as we get to these steeper areas that we're coming up.
Yeah, we're getting pretty close to that second waypoint where we're going to jog over. I'm not sure what the orientation there is. Probably a little bit north. I think that's kind of looking west-southwest, that view.
Yeah, so it looks like kind of northish. Jogs north.
I see. Right. Yeah, we're on the southwest slope. Yep. Okay.
Jogs north, then we'll kind of
Okay.
northeast. Yep, no worries. Thank you. And if you're just joining us, you might see a blue screen ahead of you of water column. We were just diagnosing some propulsion, not issues, but maintenance. And we are back on board. Everything is going well. We came off the bottom just for a little bit for safety reasons while we switch over to a different propulsion system. And that's purely just so we have situational awareness of where we are relative to the bottom while we do that switch over in propulsion of the ship. That switchover has been made. Everything is good, and we are going to be going back down to continue our dive along this Guyot and see some of the more of the amazing benthic epifauna that we've been observing. We're slowly continuing our transect up one of the lower pinnacles offshore of this Guyot. We started at a maximum depth of about 1,500 meters below sea level, and we're hopefully going to try and shoot to come up to around 1,340 meters. We're at about 1,410 right now. We're at a latitude of 39.373 north and 31.844 west longitude. We're about 54 kilometers west of the island of Flores. That's one of the northwestern islands of the Azores archipelago, Flores and Corvo Island. Now, it's interesting where we are because for the last few weeks we've been diving on areas of the mid-Atlantic ridge or off of the ridge axis. I think as far away as we got was one of our last dives, which was about 30 kilometers west, 40 kilometers west of the ridge axis on crust that was about 2.2 million years old, and we dove on a submarine volcano that had been bisected by a normal fault in that area. Now, where we are, we're kind of getting into this area. We're in the confluence of both the Mid-Atlantic Ridge, but also hotspot volcanism that formed the Azores Plateau. Now, there's a little bit of a debate in the scientific community about how hot that hotspot actually is. Is it really driven by a thermal input, or is it more water-rich material in the mantle that's causing some type of thermal buoyancy? What's interesting to me as a geologist is that the crust we're on that was formed through mid-ocean ridge volcanism, and that's from the Mid-Atlantic Ridge, the crust we're on is about 9 to 10 million years old, probably. The Flores is on crust that's about 9 to 10 million years old. But Flores Island is really only about 2.2 million years old at the oldest in terms of when it first started submarine volcanism. The subaerial portion of it is significantly younger than that. And so, being 50 kilometers west, we're on even older crust, probably about almost 14 million years old, but it's unclear to me the volcanism that likely forms the base of this Guyot, the seamount underneath it, how old that might be.
I saw while I was eating my lunch, you were looking at some pretty cool jellyfish there.
Oh yeah, that was 100% not me. That was our amazing crack ROV team who was troubleshooting and multitasking and looking at beautiful jellyfish and our video engineer.
I also noticed that I think both pilot and video noted that jellyfish with the little nubbin on top had not been seen before, and good eye to them. And I realized that I should be asking you in the front row more often, is this a new coral? Because they see more than we do.
Well, they certainly see way more than I ever do.
Yep.
Always willing to share our opinions.
Thank you.
Our technical opinions on what species it is.
They're welcome. We have not collected any rock samples thus far. Yeah, we'd like to get over to waypoint 3. The slope should be steeper over there. And yes, thank you. I'm seeing a couple of Chrysogorgia colonies down here, and what I think are probably Primnoidae. Pilot, if you're able to look at that fish, I don't know if we're stable yet. But upper left, eel-like one. Thank you. We heard from Ken Sulak earlier today that so far on the previous 8 dives, we haven't seen any synaphobranchid eels, which are the cutthroat eels, or halosaurs, and those are very common deep-sea eels that we frequently see. So it was a little bit surprising that we hadn't seen any on any dives, and yet this morning, shortly after we arrived at the bottom, we saw both species, and I believe this is a synaphobranchid eel, cutthroat eel. I don't think we've seen this large yellowfin lower right, so maybe just a quick look at that. So this close-up, we are looking at a plexaurid, family Plexauridae. It's an Octa Coral, and I find it kind of interesting that I'm not seeing any large snake stars in the branches. Can't say for certain what genus this is. I see several barnacles in the branches, the Crinoid, the suspension-feeding echinoderm. This could be something like Paramyrcia, Placogorgia, Muracetes. All of these plexaurids are a little bit difficult to distinguish in situ if you don't have samples and previous knowledge of the biodiversity of an area. But these are all reasonable suggestions that I mentioned. Lots of amphipods also, these small little crustaceans. They're kind of pink, and you see their white eyes are reflecting the light. Another crinoid. And as we zoom out there, you see that there's this amphitheater sponge underneath with lots of crinoids on it. There was a hermit crab in there. That's good for me, pilot, when video is complete. All right, sorry. There's all kinds of things going on here in the control room. Now, one thing, and I apologize if anybody was trying to get me. I was off the headset talking to our EC. There's still all kinds of crazy interest in those holes. So one of the things I'm seeing here, I think will really get Ashton excited when he comes back, because he was hoping that we might see some structures that look pillow-like, and this is pretty clearly pillows to me, I think. If we can look at the upper right, are we in a position or are we still working on the camera? Okay. Upper right looks like the end of a pillow is what I'm looking at just to see. Yeah, they're so encrusted, I don't know that we'll see anything. If, you know, what I'm learning from Ashton is if they're broken off at the end, you can see into the interior and learn a little bit more about their formation. He's said a few times that these are so old relative to the ones that we saw when we were right in the axial valley that not sure how much we can learn, but we'll try to get a good look here. There's somebody stuck on the inside there. It looks like I see a series of legs. I'm kind of thinking it might be an isopod. Are we at max zoom? Yeah, let's go into that crack. I see what I think are long antennae and legs, and I don't think it's a shrimp. That's good. Huh. So whatever it is, you can see actually farther back, there's another one. My suspicion is that those are isopods. And once in a while, we see them sort of, these ones with the really long legs swimming in the water column. Yep. The one at the top back is a better view. So that's almost surely what they are, munidopsid isopods. And sometimes referred to as the water walkers when you see them paddling up in the water column. That's a cool view. They're really tucked into this little cave-like feature. Oh, good. Ashton's back. We've found you a whole bunch of pillows. So why don't we zoom out so Ashton can see the general scene here. I thought what I was seeing here might have been a cut-off open pillow, and we're actually looking inside of it. But it's obviously pretty old.
Oh, yeah. Wow. Yeah, so you can see that outer surface of it on the side closest to us that looks much more kind of pockmarked, right? And then there's a crust above that. Yeah. And so that, to me, looks like that outer glassy surface that's weathered off and kind of fragmented off. And then the upper portion is probably a lot of manganese precipitation.
So all of this, you're saying that layer that's just back from the
I think
edge?
It's a mix of that outer glassy rind and then manganese precipitation on top of that. You know, if you zoom in even finer on that black outer rind, you know, you might see kind of this granular sandy texture, and that's kind of indicative of some of the manganese precipitation that we see. It doesn't look like that glassy fragmented surface that we saw on the younger pillows.
And would that that texture you're talking about be because as the precipitation is occurring, it's also incorporating some of the particles that are sinking?
Yeah, and I think also just it's just the way that it precipitates out. You know, it kind of it forms kind of just little individual grains until they kind of precipitate into a full surface. But you know, that was one of the questions I was really curious about, given the age of the area we're in. What is going to be the likelihood of seeing pillows? You know, so this makes me think that, you know, the fact that we have these pillows exposed and they're not under 10 meters of sediment or, you know, 20 meters of sediment makes me feel like the volcanism that is actually responsible for these pillows is probably much more similar to the volcanism that created the islands of Flores. So it's much more hotspot driven and not necessarily mid-Atlantic ridge volcanism. You know, as I said before, where we are, we're probably on 12 to 13-year-old oceanic basement crust that was formed during the mid-Atlantic ridge, but maybe 2 to 3 million-year-old hotspot volcanism.
Right. Getting a really nice view of this shrimp here. Looks like it's doing a little bit of maintenance on its head. This is a nematocarcinid, and that name translates to basically needle leg shrimp, and you can see why it gets that name. Very delicate legs, very long legs, and a key is that it's got these really long antennae that are curling back. Those are sensory to presumably detect prey in the water column. There's our synaphobranchid eel, the cutthroat eel, moving past again. A magnificent sponge off to the left. Or a stocked crinoid or sea lily in the background.
Now, this is one of those areas that can be really hard to get a good sample for kind of the igneous petrology, the original mafic material. You can see there's a lot of that coral and carbonate in that little interstitial area between the pillows, so you don't see the opportunity for lots of fragments.
Yeah.
And then you can imagine there's so much weathering and manganese incrustation. Trying to see an area where you want that outer glassy rind is difficult.
Yeah, I think that one we zoomed in on was a good indication of the layering that's going on, and how you can be fooled if you don't see that from a non-specialist, I mean. That we're seeing this original basalt. That's something I learned pretty early on, I guess, in my deep-sea diving, was that unless we were right in a rift valley, we probably weren't seeing that nice original pillow, or maybe in a volcano like in Hawaii.
Yeah.
You know, there you can be assured you're seeing something a lot fresher. I said needle leg. Mike Vecchione points out thread-legged shrimp. Really nice view here of some of the diversity on this raised topography, various kinds of sponges, both glass sponges and demo sponges. I see black coral, that's that thing that's kind of brownish red in the upper right corner, and part of the skeleton looks like it's completely overgrown by barnacles. You can see those large white spots on them. Barnacle larvae will be swimming around the water column looking for some suitable habitat on which to settle and grow, and very often that's some bared skeleton of a coral. I think there's a barrel sponge right above that very bright white sponge.
The one with a little hole in it?
They got a little hole in it. That might be the genus Geodia. I'm seeing, I believe, an Iridogorgia coral down below, the spiraling Octa Coral.
Now, I feel like here we've definitely seen sponges that are larger than places we've seen previously. Is that a fair assessment?
Certainly when we were really deep, the sponge fauna there was dominated by really small stuff. It's probably a good reflection of the food supply. The sponges are animals, just like all these corals that you're seeing are animals. They are all requiring food in the water and to consume. If you don't get enough energy, then you can't add to your tissue and grow large. And so, on average, in the deep sea, the bulk of animals are actually quite small, and there's a lot more stuff that we're not seeing that's hidden in the sediments, or is tucked away in the crevices, or is crawling through the interstices of these branches of the corals and so on. We're just sort of getting the megafauna, and that's because there's relatively little food, and especially once you get down to abyssal depths. I think you said it really well yesterday, or no, it was when we were 2 days ago when we were in that Rift Valley at 3,300 meters, that we still weren't even at the average depth of the Atlantic. I mean, the ocean is really deep. The ocean gets down to almost 11 kilometers deep. Much of it is abyssal basin at 4,000 to 5,000 meters deep. And there are thin slivers, the trenches, the subduction zones, where there's plates that we're seeing born here on the Mid-Atlantic Ridge sort of go to die and get recycled into the Earth's interior.
Yeah, I think, what did we say? The average depth of the Atlantic is about 38?
38 is what you said. I have this number from a paper, was it Chalumet, I think, 3682 meters. And one of the things I thought that was so interesting about when I saw that paper was realizing that we're still trying to estimate that average depth, and it's not because the ocean depth is changing, but it's because we're getting better and better maps of the seafloor. And the more of these, and this expedition is a great example. Shannon Hoy has just set down our mapping lead, and there's just the stunning maps that get created every night. We get up in the morning, and I can't believe some of the new things that we found, or not that we found, but that they've found and mapped. And so every time we find and can map well a new seamount, that makes the average depth a little shallower, because we have a little more of the seafloor that's peaking up. And so it's the discovery of seamounts, the better mapping, that is changing our estimate of what the average depth is.
Yeah, so I've always found it interesting, the way a lot of these seamounts are originally mapped is actually by looking at satellite altimetry. So
that is so cool. It just blew my mind when I first heard of that.
Yeah, so you can think of the ocean surface as a what we call an equipotential surface. So it follows an equipotential curve in the gravitational force. And that means that if you have a seamount in the ocean, at the ocean floor, then the actual the ocean surface bulges a little bit, and we can follow that bulge by bouncing a laser from a satellite off of the ocean surface, and we can infer that there's a seamount beneath us. But that same effect can also be caused by a density increase in the oceanic crust, so there doesn't have to be a seamount there.
Right.
So, you know, you have these estimates of where seamounts are, their sizes of them, but you don't really know until you go out there because there's these crustal and density influences.
Sorry, we were just zoomed in on this really large sponge that is tipped over. I think it's a dead sponge, but you could imagine this is another sort of big radar dish sponge that would have been upright at one time. And it doesn't seem to have as many organisms living on it, and that might be because it's no longer standing up. It's not in a higher current flow. We talked about this a lot yesterday, that the skeletons of these sponges are made of silica, and that silica will persist here on the deep sea floor much longer than the live cells. The cells will be getting broken down, process of decomposition, just like on land. There are bacteria. We don't talk about them very often, but there are fungi that live in the deep sea, and there's not much known about them. I'm kind of intrigued by this little cluster of polyps that's on the upper right of the screen. Those are, looks like those are Octa Coral polyps, and I think these are the ones I mentioned are in the group, the Cornularidae. And what's different about them is that they have these little exoskeletons at the base of them. And so it's not just that they grow as a mat or a ribbon, they do, but they are also creating this exoskeleton that I'm pretty sure is using the molecule chitin, which is the same molecule that forms a lot of the exoskeleton of, for example, that shrimp up there. These corals can do wondrous things.
And so just to summarize, because I've forgotten already, what have we sampled so far on today's dive?
On today's dive, this sheet is not going to tell me what we've sampled, but this sheet will. We have a variety of things in front of Ashton and I. It gets kind of overwhelming sometimes with all the different computer screens and computers and access to information. And one of the things I'm doing right now is I'm looking at a spreadsheet that I've just downloaded from a database that we keep. So every time we take a sample, either a water sample, a rock sample, a biological sample, we enter it into this database, and it gets stored in a spreadsheet, and then I download the spreadsheet at the end of the day, and we go into the wet lab and process the samples. So when we first hit the floor, one of the first things we were so surprised to see and kind of astonished was these lines of holes, the same sort of mystery holes that everybody's been kind of talking about and coming up with hypotheses for what caused them. And so we saw those holes, so we stuck our suction sampler into one of the holes and sucked up a bunch of sediment. We're hoping that if there is some organism down there that's creating the holes, maybe we got it sucked up. We then clipped a piece of some sponge, not the sponge that we're looking at here, but another one that was very frilly in appearance, and the folks on shore did not know what species it was, but they had noted it before and wanted to get a sample so we could put a name to it. And then we sampled one of these yellow-brown octocorals called Acanthogorgia. In fact, right on cue, that's an Acanthogorgia on the sponge, far right. You can sort of see that yellow-brown color. So we sampled one of those, and it was actually on a skeleton, almost exactly like what you're seeing here, a dead stony coral. And it had 2 or 3 other corals on it, so I won't know what those are until we get them up. And then we've taken several water samples, and we filter the water through a very fine mesh up here on deck, and we'll take those back to the lab at the Smithsonian Institution, and they will be examined for all the DNA that's in the water. All of these organisms that you're looking at here are shedding their DNA into the water column. And so we can capture that, and using new methods of next-generation sequencing, get all the sequences, do a bunch of bioinformatics, and then get a sense of what's the diversity in the water column here or coming off the bottom, and in some cases even identify it if there's a match in our database. And in fact, as I'm looking at this scene and I'm seeing so many different corals and sponges here, I realize that this is a great time to take Niskin sample number 3. Pilot, do you copy? So mounted on the back of the ROV are 5 water sampling tubes. It fired. Thank you, Caitlin. Video clipper is just pointing out to me that our water bottle just fired, and so I'm making the addition to the sample collection sheet. So yeah, we have 5 water bottles on the back of the ROV that we can trigger at whatever depth that we want. And so we usually take one on the way down in the water column in what's called the deep scattering layer. This is where our sonars tell us the concentration of midwater animals are. That would be both fish and zooplankton. So getting a water sample there can help us understand who is in that concentrated layer of midwater animals. Then we take another one as soon as we get onto the bottom. Then we take one at an arbitrary place that we find interesting. So I thought there's so much diversity here. Let's trigger it and see if we can pick up some of that diversity in the sample. We'll take another one just as we leave bottom, and then a 5th one when we go back up through the deep scattering layer. I'll bet that was a longer answer to your question than you thought was coming.
I don't have to say that, but what I was going to say was I said that answer is so long I forgot what the question was.
What samples have we collected?
What samples, that's right.
And I thought you were so interested
I was.
in that question.
I was, but then we got sidetracked because we started taking another sample.
Pilot, can we zoom in on the this feature that's in front of us? There's a clump on the upper right, and there's a fan sticking off the left of it. Thank you. I think that's a bamboo coral. No, the one right in front of you, just above the, I don't want to use colors. Exactly. So below the lasers. Yeah, well, but I know where they'd be. So that thing that's just to the right, that fan. And I think it might be the same as the large fan that I think your eyes were drawn to at the top of the screen, off the screen now. But let's just get a good look. And there's so for context, the thing with these sort of teardrop-shaped polyps on the upper right, that's a Chrysogorgia colony. As we're zooming in, yeah, this is going to be a bamboo coral. If you look at the lower part of the skeleton, you're going to see that there are white chunks to the skeleton that are interrupted by black lines. The black lines are protein-based nodes. The white are the internodes. And once again, as I look at this tissue on this coral, it's really reflecting the light, and it's not allowing me to see through to the skeleton. Perhaps later we'll see some other bamboo corals and you'll see what I mean. You can see all the way through the tissue and you can very clearly see the skeleton. So that reflectance is already telling me that in the tissue are lots of very small calcium carbonate structures that are reflecting the light and making it opaque. So I already have a good sense that this is probably in the Jasonisis group of bamboo corals. Thank you. So I just got logged out of the chat room there briefly, but I'm back in the chat room, and I see that Jean-Marc Gagnon, who's the curator at the Canadian Museum of Nature, who has been actively working on the giant file clam, and we saw one of them earlier. I keep looking at Jean-Marc to see if we can find another one, but he says that the eDNA samples will be great to see if we have these giant file clams around. And if we do find it, then we might be able to use that DNA to say, well, is it the European species, Acesta excavata, or the Northwest Atlantic species, Acesta cryptodelphi? And that brings up this interesting question that one of the major goals of our program is to provide the observations and some samples and data to understand the connectivity across the North Atlantic Ocean. And so the eDNA samples are going to be part of that, because we can identify, in some cases, down to species. We can only identify, I want to make this clear, we can only use eDNA to identify down to a species name, that is to connect the name to the DNA, if somebody has already identified a species, sequenced that species, and added the name to the database. It's a comparative sort of operation. Independent of that, what we can do with the eDNA is say, we have this many different pieces of DNA in our sample. And so we can say there are probably this many different species. We just might not be able to name them all. But that's also a lot of use, because let's say that we have had these dives down to 3,000 meters and 800 meters and 1,200 meters, and we're going to different parts of the ocean. And one of those dives revealed that when we took the eDNA there, we found very different sequences than we have anywhere else, and we can't identify most of it. Well, now we know we should use our resources to try to get back to that area to try to understand a little bit more about the diversity. So lots of uses for the eDNA.
There's another barrel sponge.
Yeah, it's an interesting color, isn't it? I was starting to doubt myself the last time that, well, maybe it's an anemone that's really closed up and pulled in the tentacles, but no, I think it's a barrel sponge.
So you talked about the integrated research and the connectivity of these systems.
Right. So when we moved the Okeanos Explorer, the ship, from the Pacific to the Atlantic, we were looking at a way to pull together what are all the different needs that exploration could help provide some data to answer. And the program overall, hang on just a second. Sorry about that, folks. It's a safety drill day, so we're just taking it off the air while there are some potential alarms going on. Yeah, please zoom on the fish. Oh, this is different. This is definitely not a cyclothonia. This, to me, looks like a baby rat tail.
I was going to say a rat tail.
Yeah, I don't know that it is, but that's kind of interesting. Really quite small. Very dark. We were talking about this yesterday. It kind of looks like a large head with a tail, and it's kind of missing the rest of its body. So I apologize. To get back to what we were saying, we're doing these explorations in the North Atlantic, and we've been doing this since the fall of 2017, and so we've had this program going on now for just over 3 years. And the name of the overall campaign is ASPIRE, and it stands for the Atlantic Seafloor Partnership for Integrated Research and Exploration. And the idea was that we would take input from interested marine scientists and managers, conservation folks, fisheries folks in the North America, United States, and Canada, but also on the other side of the pond, from Europe, from Africa, to all the people who are using the North Atlantic. There's a lot of shipping traffic, there's a lot of commercial interests, there's a lot of fisheries interests, and there's a lot of scientific interests. And we sort of bound that all up together and looked for what are the best ways that we can use these assets to provide you with some first look characterizations of some of the habitats that you're interested in seeing. And I'll take a break from that to have a look at this exquisite sea star. This might be a goniasterid as well. I'll go out on a limb and say that. And perhaps we'll hear from our resident expert. Well, I'm sorry, he's not our resident expert, but Chris Ma does like to participate a lot in our dives. He's at the Smithsonian Institution.
What makes you think it's a goniasterid?
Yeah, I'm looking at, you see outside of the arms, there are some rather large plates that line those arms. And my familiarity with that family is that they tend to have these oversized plates there. But I could be wrong. Now, yesterday we collected one. I call it the cookie star. cookie cutter star. There we go.
Chris,
where the arms are greatly reduced. And
Chris Moss says Circaster or Circaster?
Circaster. So he's giving me the
yeah.
name of a genus.
Yeah.
I was giving you the name of a family.
Okay.
So that's great. Circaster. So his identification is at a more fine scale level. Yeah, yesterday the one we collected didn't have those long arms. It seemed attenuated, let's say.
Oh.
Hey, this is Chris Moss.
Hey, Chris.
Hey, I'm like that bad legend about saying Bloody Mary into a mirror. This is actually kind of interesting because it is Circaster, but I've never known Circaster to occur out this far, especially not on the European side. I realize that we're not quite there, but Circaster americanus is called that because it's known to be something which lives sort of on the western tropical Atlantic side. So this is definitely a new record if it is indeed, I don't know if it's americanus or not, but it's certainly got the large arm plates as Circaster. If we can afford to collect this, it would be worth documenting. It's a new occurrence. It might be feeding on some of these corals and other sponges in the area. Scott, you actually identified Circaster americanus as feeding on that large Paragorgia several years ago and came upon the most amazing discovery that some of those sea stars were still on that coral, feeding on it a year after it was first discovered.
18 months,
which says a lot about the yes, and so and that was a mind-blowing revelation as far as I'm concerned, because I would have easily attributed weeks or months, but for it to be out there for almost a year, feeding on the same things, and I realize I don't have conclusive evidence that the 2 or 3 individuals that were on that coral, that fallen coral, were exactly the same ones, but it sure did look like it. And,
I agree with you.
It is a very plausible notion that given the rate of metabolism and everything, that that they just take a very long time, and that feeding could literally take a year, especially if you've got a stable food source like that. So and who knows what all of these. So the Goniastridae, I may have mentioned this before, but they're the most diverse group of sea stars. There's something like 65, I think, with my work, probably closer to 70 to 80 genera, and there's something close to 270, 280 species. And I realize that's piffling compared to things like brittle stars or insects, but compared to the number of things in the sort of world of marine animals, that's for big animals, that's pretty good. And so that's definitely every time, like I've sort of harped on before, the last 2 times that we collected goniasterids from the Atlantic, they turned out to be new species, and in one case, new genus.
The one we collected yesterday, Chris.
Certainly.
I was just going to say, it came up in great shape.
Oh, lovely. I've been sort of slobbering over that picture and looking forward to getting it, because I'm pretty sure that's. I mean, it might be something called Astroceremus, but I also know that that's far and away not something that occurs in this area. So it might be something completely different. A close-up is usually the best way to tell. I mean, a specimen in hand.
Can I interrupt you for a second, Chris?
There's a lot of. Oh, I'm sorry.
Yep, just for a second, stand by. So just because we're getting this really nice zoom right now on what looks like an anemone from a gross level, but I actually believe is a Corallomorpharian, and we really don't see very many of these, so I just want to make a note of it. So a Corallomorpharian is another kind of hexacoral, belongs to that same group, but it's in its own order, where the stony corals are one order, Scleractinia, and the black corals are another order, Antipatheria. So this is the Corallomorpharia. They are kind of like the polyps of a stony coral, but they don't have a skeleton associated with them. And one of the things you can see is they have this very transparent tissue, and so we can see through quite nicely into the body. I can actually see the pharynx extending down from the mouth, which is that little darker brown line that you see, that slit. So the sort of quote-unquote lips are tightly pressed together right now. And then you see all the little spotting on the tentacles. Those are bundles of nematocytes, and so those would be the stinging cells that they would use to subdue their prey. And then a lot of people look at the bulbous tips of these tentacles and use that to identify corallomorpharians. And it is true that most corallomorpharians have those bulbous tips, but there are also some anemones that have those tips, so we can't just use that. But nice view. We don't see enough of these. They're really hard to identify from video or imagery. You have to collect them, and basically you have to do histology on the tissues on the inside of that gastrovascular cavity, the musculature, the arrangement of muscles, in order to identify them. It's that hard. It's a very specialized field. Thank you, Pilot. Yeah, go ahead, Chris. You can continue.
I don't know. I mean, there's a ton of stuff we don't know about them, and considering how many of them we encounter sea stars in the Goniastridae, they're probably doing something important down here that we haven't quite figured out yet, most of which seems to be predation on your things, on corals and sponges.
You call that important, huh?
I apologize for that, but I didn't know what it was. I didn't know what they were doing when I decided to study them. They just kind of started doing it after. I mean, this is literally something, this whole paradigm of sea stars feeding mostly on corals and sponges. I mean, there are, of course, examples, well-known examples from shallow water, but the fact that most of the Goniastridae were largely unknown from sometimes just single specimens was throughout most of the 20th century just sort of one of those things. Like Evoplasoma, you know. Evoplasoma was just considered this weird, rare Goniastrid. There were literally 6 specimens known, I think, or in 1997 or so. There were only 6 specimens, and each one was its own separate species. And, you know, now I mean, after the the capstone expeditions, they're everywhere. I mean, they're not common or even, you know, abundant by any means, but we see them everywhere, and they're feeding on corals. We didn't know what they did. You know, I mean, we just had them collected from. I mean, we had one specimen collected from a trawl, and you know, and it wasn't in great shape to begin with. So, you know, there's so much that's unlocked by, you know, I mean, a lot of what we do seems, I don't want to say trivial, but like the start of all of what we do begins with naming these things and figuring out what they are. That's what you do. That's what I do, and you know, and then we can, you know, we kind of like, that's the start of our knowledge. So just describing or finding these new species is then like, oh, okay, so now we know what it is. What does it do, and why is it quote unquote important? And so
I would back it up from there, Chris, by saying it's even the observation stage, right? And so I think you said before we even get to name it. And so you said something in there. I want to make sure that the audience doesn't overlook this. And that is, it's really because we've had this Okeanos Explorer observations exploring all these different parts of the deep ocean that you can now say with some confidence, this is not uncommon. We see this kind of predation going on. We see these sea stars associated with corals and sponges and so on and so forth. We've made so many observations, and I agree with you, so many things that I didn't know about or maybe had a suspicion but I'd never seen before. We're now seeing with some regularity. So I think we're starting a little less to use the word rare because we now have the ability to have the observations.
Yes. Well, someone warned me many years ago that the better term is rarely seen or rarely encountered.
Exactly.
And you know, rare is something that, yeah, I totally agree. But you know, like this, you know, but we're also seeing. Well, I mean, and yes, I you know, I mean, I've made a point several times that, you know, in agreement with you that essentially there's a ton of these observations that are almost more important than like physically collecting the specimen itself, because we can, you know, we can, you know, the value of watching something feed to me far exceeds, you know, like yet another specimen of what could be a similar but known species, you know. So
hang on, Chris,
you know, and, like,
just
for
for
example,
a second.
watching all of
Just for a second, folks who are watching this on shore, go over to camera 2, and there's just a marvelous view of the D2 hovering above that feature that we were just zoomed in on. So the distance between Sirius and D2 isn't too far right now, and it's really well lit. And one of the beauties of us sitting in this control room is we can see all these different views at the same time. We just got a myriad of monitors in front of us. So it allows us to see the D2 in context of a wider environment and to understand the organisms in a sense of this wider environment. So, for example, that one little topographic high we were seeing, is it on a slope or is it on a more flat plane? This looks like we're getting onto this flat ledge here. So just terrific. All right, Chris, sorry.
Oh, no problem. I think I was pretty much just closing up shop. I mean, in the sense that the thing that I did want to mention, though, was that one of the things that is most remarkable, and it's not even about the animals that I study, but about the brittle stars, because, you know, thanks to the very eagle-eyed observers on the ship, we've seen, I think, half a dozen weird little brittle stars that have never been seen alive before. And it really astonishes me because, you know, no one – these things come up as essentially little puzzles, you know, in trawls, and no one knows what they do. And then, you know, and then we see them here on the bottom sometimes, and they're, you know, they're dime-sized. So it really is an accomplishment sometimes to when, you know, we see these rare little brittle stars like at 3,000-meter depths on the bottom of the seafloor, and, you know, and there it is alive, and it pretty much looks like what the specimen looks like, and it's doing, you know, and it's doing what it's doing, and you know, maybe it's, you know, be nice if it was feeding, but you know, but that's what I mean is it's like, well, you know, that's this basic aspect of its biology is you know, maybe it's not all that exciting immediately, but it's definitely like the first step. So
absolutely,
and,
putting
know,
it into context of its habitat.
I'll tell you. Yes, exactly. And you know, sometimes I'll tell you, even, you know, just like that starfish that we just saw, the Cerciaster thing, you know, I mean, I'm not one to describe a species based on purely imagery and video, but the video on these things is so amazing that there's sometimes details that are enough to at least describe, you know, plausibly, you know, in a scientific journal. I mean, the specimen is always an important piece of evidence in establishing a species and what it can do, and, you know, extraction of DNA and looking at other pieces is important, but there's a lot of value in the video that we're learning now that, you know, just looking at the surface plates and such. I mean, in some cases, it's almost as good as looking at just fossil material, because there's not much left of a fossil other than the surface. So it really is just, you know, we live in a very strange and bizarre, and I would say wondrous age. So anyway, and I could go on, but I'm sure we need to go and do other stuff. So I see that the Cerciaster is pried between two rocks, so we're not getting onto that now. So I'm looking forward to the next, and, you know, and honestly, there's still a couple of deep sea, like, target species that I was expecting to see that we haven't seen yet. And as I said earlier, one of the weird things is that we haven't seen a single Brisingid since we started this.
I agree. We still have not. That is just mind-blowing.
Yeah, it's really strange. And I know the deep sea is patchy, but that's really kind of odd. But maybe it's just a combination of current and lack of food.
Maybe it's upslope here.
Have fun looking for coral.
Thank you, Chris.
Yes.
Thanks for your comments, as always.
Thank you. Bye-bye. Oh, always fun to call in. Thank you. Bye-bye.
Go ahead, Nev. Copy that. We're seeing plenty in front of us, so I think that's perfectly suitable. Thanks for the heads up. Yeah, something that Nat just said is we can clearly see the slope is getting steeper. We've seen, since we've come back from lunch, there's a lot more pillows that have been exposed, more hard substrate. We went through a patch there where the ridge that we were on, the slope was much flatter. I was trying to think of it. Let's see. The slope would be, and I'm going with the numbers in my head.
Lower?
Lower, yeah,
Yes.
okay. So anyway, it's flatter and more sedimented, and we weren't seeing quite as many topographic highs that were supporting these suspension-feeding organisms. If we could have a look at the two fans that are on the lower left right now, very light in color. I think that these are primnoids, and we haven't had a good look at the primnoid fans today, so let's see if we can get that. Just to the left of center, I think. I don't see the lasers on, so not sure. There we go. Yeah, lasers just above it. Yep, thank you. I think that yellow sponge back there, lower right, is Hertwiggia. I've learned that now. Lower left, I'm sorry, is Hertwiggia. Yeah, this is sure looking like a primnoid to me, so that means the family Primnoidae. It's kind of Octa Coral. Primnoidae is one of the three major diverse families in the deep sea. Oh, no way. There's a Brisingid. Right after Chris hung up, our first one of the expedition. All right, first let's look at the corals, because I know I'm not going to get a word in when Chris calls. Coral first. Can you feel? That's Chris's fingers hitting the phone.
Yeah, hitting the phone.
Yeah. So this is definitely Primnoidae. I'm kind of looking at the branches. One of the things for those of you I'm sure wonder, well, how do I come up with these names? I'm looking at the polyps. I can see the polyps are oriented in these whorls, meaning they're coming off from a common point. There's like 3 or 4 that go in a circle. They're oriented downwards. They look kind of stiff, and I can see enough that I see little rings along the body, and those are telling me where there are scales that are overlapping one another, and that's a hallmark of the Primnoidae. So that's pretty good. And then feel free to drift up to the right, and we'll get on that Brisingid. This is another kind of sea star that has a specialized mode of feeding. It's actually perfect that it's right next to a crinoid. These are two different classes of echinoderm. One's in the crinoidea, one's in the asteroidea. And they both are feeding the same way, these two species, in that they are putting their arms up in the water column to feed.
I'm back.
All right, there you go. I knew it. Hey, isn't that cool? You just say the word and there it appears. Ashton tried that yesterday with hot chocolate and cake, but it never materialized. But it worked with the Brisingid. So go for it, Chris.
Well,
we're going to zoom in here for you just a moment.
Thank you. This was the Christmas curse during the time that I was on the ship, was that every time I walked away from the screen, which was literally almost every time I went to lunch or whatever, that's when they would see sea stars. Because when I was there, it would just be hours of corals and rocks. And then the first time we got
that sounds beautiful.
Every time they would see like sea stars, I would be at lunch or in the bathroom or some kind of nonsense like that, and so it would be, and in fact, this almost seems to be the case here. Every time I walk out of my office away from the screen, it's like, oh, there's a sea star. It's probably a new genus and species, but Chris is downstairs in the bathroom or doing something. So, but there's definitely, and there's definitely it's weird sometimes how they just don't, you don't see any for just a long time, and then all of a sudden, well, it's like buses, you see, sometimes there's nothing for an hour, and then there's 3 of them all at once.
You noted before that maybe we weren't seeing it because there wasn't enough productivity, but there's plenty of productivity that we've been noticing. There's lots of marine snow, and densities and sizes of a lot of stuff that we've been seeing is large. So I wouldn't say that's the answer for why we haven't seen a Brisingid, but finally we are seeing one here. And Chris, just before you dialed in, what I was comparing to was to the crinoid just to its left, and noting that they're both suspension feeders in different classes of Echinodermata, but they're capturing their food in really different ways, if you want to explain that a little bit.
Yeah, and that's actually really interesting because so Brisingid are sea stars that, if everybody is looking, so all of those arms have spines on them, and those spines are all covered in a sock. Each one of those socks is imbued with many, many small little bear claws, and those little bear claws are called pedicelaria. The pedicelaria, as I've euphemistically called them, act as kind of like this whole thing acts as kind of a Velcro of death. Small crustaceans swim through these spines and the animal's arms, and they're caught by them. And, and most of the things that are documented have been crustaceans, small amphipods, et cetera, et cetera. But I think years ago, during one of the Atlantic cruises up on the coast, we actually saw a little bristlemouth, a cyclothone, or cyclothoe, get caught in the spines of one of these, and it couldn't get loose. And I was really impressed by like how like if these things were bigger, they'd just be terrifying. But they can capture small, amphipods. I've looked at really nice collections of these individuals, and I've found like all of the little crustaceans in the tube foot grooves, and it was all worked out in the 80s and the somewhat simultaneously by Craig Young and Roland Dempson, but also by the Russians, both of whom did sort of work that was good in its different groups of Brisingids. But basically the food gets caught and then gets moved via tube feet down to the mouth. So the mouth is facing down. Now, this is in contrast to the feather stars, which we see almost all over the place. And what's really kind of amazing, and there's this phenomena in evolution known as convergence, and that's basically like how the physical structure of something, if it has the same function, it arrives at the same structure. And that's what you get with Brisingids and feather stars. You know, the way that the spines are arranged and the way that they're shaped. And in both Brisingids and feather stars, there's some differences, the pedicelaria and the fact that sea stars are actually predators. Oh, and there's another sea star down on the rock. I'm having a good day today. But like feather stars, even though the functional structure is different, the overall shape is the same. They have the what's called the pinnules, the feeding bits that come off of the arm. Are structurally arranged in a similar fashion, and they take advantage of the same kind of hydrodynamic regime that that they use to pick food out of the water. And so it really is amazing to see just how similar they are, in spite of being divergent for so long geologically. Okay, well that. Chances are that thing that we just visualized is something that we won't be able to get and is probably new. So, I'll just kind of leave that there. I wasn't able to sort of get a real good hang on what it looked like, but
it's
that's
pretty
fine. It
pretty far.
all the time.
Yeah, it's pretty far tucked in there, Chris. And we got to leave something for the next generation of sea star biologists. Huh?
I totally don't know. Yeah, no, I totally get it. Trust me, there's, although I'm always after the bit to see something new on these cruises and to collect everything that is new, the truth is that I have plenty to do. And, and there's a long queue of new species that I'm currently describing. There is the ones that are from the last cruise, but also there's a bunch of things from, for example, an Antarctic cruise in 1966 that I'm looking at. There's material from museums all over the world. I'm going up to the MCZ, to the Museum of Comparative Zoology next week to look at new things that were collected, not necessarily new species, but new material collected by the Nautilus. And so, I'm one of the few people that actually publishes on the taxonomy of new sea stars, living sea stars. And so, I have a lot of of stuff that that is backed up essentially. So, and I get to pick and choose at the moment, but it is, I tend to focus on the deep sea thing since it requires a broader view, and there's some very interesting things. But I mean, I've been sent material, pictures, for example, from the Western Australian Museum, and they have amazing things that were recently observed and collected by the Falcor. So, the world is my oyster, at least for now, until the next young person comes up to inherit and share all of this fun responsibility. I would also say, just as a shout out for my organization, we talk a lot about the repositories and where all of this stuff goes, and the Smithsonian is the repository for things collected by the Okeanos Explorer program, and so materials that are collected here come to the museum and are processed by our staff and are made available to the scientific community, by request on loan. And so, this material is held in stewardship for the public, for anyone who, is interested in the material, but of course, responsibly. But it's available to scientists, and if you have an interest, you should contact the Smithsonian or the Natural History Museum. And, we have the material here indefinitely, and the average time from shelf to discovery is about 21 years, as it turns out. And so a lot of the material here, I mean, we're talking associates, we're talking anything that was collected on these cruises, and that includes any of the stuff that you pull off the rocks, is stored here and kept here forever. So, please do, I hope someone will, if you have an interest in whatever sort of small epizoic fauna lives on these rocks when you visualize them, please contact us, and, we'd be happy to help you with. Oh, wow. Oh, they're hermit crabs.
Yeah. Only the lower one's a hermit crab, Chris. The upper one, I think, is an actual snail, and I'm wondering if it's feeding on the hydroids that are growing on this crinoid stalk.
I've actually gotten on top of that observation from 2017. It was a bathicrinid stalked crinoid. We saw in 2017 during the Pacific an observation of a snail with its proboscis clamped over the anal vent of a stalked crinoid, or at least something like that. And I'm going to be following up on that to make sure that we have all of that figured out. But I've already gotten to the point where it's a bathicrinid, and we'll be we'll be trying to get more of that out as I get more. Oh wow! And the hydroids are covering up all of the top of the stalk like that. That's amazing. Yeah,
and we've seen this crinoid now many times over a few dives, and every time I've gone in for a tight shot, it's had this what I think is Hydractinia hydroid overgrowing the stalk. So that's an interesting and consistent observation. I don't think the snail looks anything like that one that you were just referring to. But all right, I think we're getting ready to make a little headway up this slope. Chris, thank you so much for those observations.
Thank you. I always appreciate when you stop to look at pentameral animals. I will say that every one of the crinoids that gets collected is appreciated by the community, the stalked crinoids, because they are so rare, or at least rarely encountered, and especially intact. And, I think every time one of these comes up, I think a good chunk of them turn out to be new, or undescribed. Don't ever think that just because we see it that, it's already known to science. Sometimes it is, and if it was that, red one from the Pacific, that's one thing. But there's definitely a lot of these weird sort of stalked crinoids that occur all around the world that, if you're looking for something to collect, pick up a stalked crinoid. It'll probably be something that we haven't encountered before.
Great,
thanks so much.
For that. Yeah,
I'll
thanks.
be back, I'm sure. Thanks. Bye-bye.
Thanks, Chris. Yeah, Chris made a really interesting point there, and that is that especially with more and more video, ROV video and drop cameras coming down, we may see organisms that we sort of check this box like, yep, that's common. We see that a lot. However, the number of collections that are made is far less. If we could go to the upper left, that line of colonies looks really interesting. So sometimes we are guilty of overlooking the common because we see it a lot, but we've never actually picked it up, and so we can't put a name to it. And this is actually similar to one of the things that our Azorean colleagues have said, why some of these collections that they're directing us or requesting that we make are useful, because they can see the stuff in the camera images that they have, but they can't always identify it.
Yeah, that was a really great description he also gave of the collaboration with the Smithsonian and the work they provide. And NOAA Ocean Exploration performs ocean exploration not just on the NOAA ship Okeanos Explorer, but also on a number of other ships. They've used the NOAA ship Pisces before. They've recently, in the last few years, done Alvin dives using the RV Atlantis. And then they also provide a federal funding opportunity to researchers to apply to do ocean exploration work on other ships of opportunity.
Right, this Okeanos Explorer is just one facet of that office. So what we're looking at here, these large orange-colored bushy-like corals that you're seeing, these are black corals in the genus Leopathes. There's a couple of known species out here, Leopathes glaberrima or expansa. If you've been on some of the earlier dives, you've heard us say that among corals that have been aged using using stable isotope data and radioisotope data, Leopathes is the oldest known coral, and in fact I believe is the oldest known animal, and it has been aged up to more than 4,200 years old, and so they're extremely slow growing. I believe I gave an estimate on the order of about 50 microns of growth per year, and I said that the average human hair has a width, let me find
75 microns.
75, thanks.
That's what I'm going to guess.
75 microns. And so, yep, you're bang on, 75 microns. And so the rate of growth of these would be such that it's 25 microns, less than 25 microns per year of the black coral. It would take 50 years or more just to grow 1 millimeter extra in diameter. So you can imagine that these corals that we're looking at are really very old. Quite impressive. Another hollowed out pillow up here, Ashton.
Yeah, and probably another little puka for any type of community that wants to.
The last one had a couple of isopods tucked in there. It looks like, talking about things that are very old, some of these corals have been in place for a very long period of time, and you can also see that because they are also kind of blackened. I think part of that is some incrustation. Part of it may be bacterial decomposition, but that's not all of it. And so who knows? Maybe some of this coral skeleton that we're seeing here is 20, 30,000 years old, and then there's live coral growing on it. Ashton, stick your hand in there.
I'd love to stick the ROV hand in there and get a sample, but I don't think any of that's going to break off.
Yep.
Yeah.
And can we get the lasers back on, please? Thank you very much. So you can see the base of that old dead coral there, the way it attaches to the edge of that pillow and has grown up, and it's been infilled by all kinds of stuff. Those laser dots are 10 centimeters apart. You can see a little bit better inside your pillow. Yeah, there's not much going on in there. Yeah, it looks pretty solid and encrusted, and so I would imagine that collapsed a very long period of time ago.
Yeah. Yeah, it could have easily been during formation. If the currents are strong enough to keep this area swept away from any of the overlying sediment deposition, this could be 2 to 3 million years old.
Yep. Interesting little urchin there on the upper lip. Okay, let's keep moving on. We're at 1,455 meters depth. When we first settled on the bottom this morning, we're at 1,498 meters depth. We're about, 50, or is it less than that? 54 kilometers west of the island of Flores on the Azores Plateau. That bright white and yellow colony that you see on the right is a stony coral, scleractinian, genus Enelopsamia. The skeleton is white and the live tissue is yellow, so it looks like parts of it are alive and parts of it are dead.
Now, sorry, I know you've probably explained this to me too many times, but I'll be the biology student. Can you explain the difference with the stony corals?
Yep. So let's do a little pyramid here. So
Yep.
at the top of the pyramid, we have the phylum Cnidaria. Within the Cnidaria, we have three classes: the Hydrozoa, the Scyphozoa, which are jellyfish, and the Anthozoa. That's not true. We actually have some others, the Cubozoa and so on, but we're not going to worry about those right now because we don't see them. Within the Anthozoa, we can divide that up into the hexacorals and the octacorals. So all the sea fans that we've been seeing, the bamboo corals, the primnoids, the sea pens, all that stuff, those are all octacorals. They all have polyps that have characteristic 8 tentacles, and that's one of the easy ways to identify them. But what's more interesting is that they're evolutionarily all link back to a single ancestor. In that other group, we have the hexacorals. That includes the sea anemones, the corallomorphs, the zoanthids, the black corals, and the stony corals. The stony corals belong to the order Scleractinia. And one of the reasons I keep saying stony coral is because on average, most people are more familiar with a stony coral. That's what builds the shallow water coral reefs that we see, those brain corals and the elkhorn corals and all that sort of stuff. There are lots of stony corals in the deep sea, but they aren't the kind that build those really solid mounding structures that build up reefs. They tend to be either solitary cup corals, we've seen several of those today, or like this branching morphology that is very, very brittle, very easy to damage. You know, if we reached out with the manipulator and tried to grab that, it would be like shattering china.
Oh, really?
Yep. And that's why you see all this rubble. It breaks really quite easily. But it is a very stiff calcium carbonate skeleton with a very thin veneer of living tissue on it. And you just see a fish, or you just go, whoop-de-doo?
The spiral.
I see, the spiral, yeah. So that's probably an Ovidogorgia, now dead, dropped all of its branches.
Okay. And there's there's a live one. There's a live one,
yeah. Good call. Yeah. Good call. On the left side, lower left, or center left, there's an alive Ovidogorgia. And the feather that you see on the far right, that's another black coral, Parantopathes. And it looks like a crinoid tucked under the lip of that rock. Yep, one of these 5-armed crinoids. So that's the one that uses basically 2 feet to capture its food and move it to the mouth, and the mouth is on the upper surface there. Different from the Brasingid that we were all excited to see a short while ago, which is a modified sea star. Sometimes I get fooled, you know, I'm looking at here and it looks like a fresh surface to me, but I imagine it's just the angle and the fact that it doesn't have sediment associated with it. It's tucked underneath, has that darker appearance. Looks like a metallic Gorgia colony, perhaps. That's hanging on the far right. Talked about that a few times. They're the ones that have their Ophiuroid associate, the brittle star associate, Ophiuchreus oedipus, And we've talked repeatedly about how amazing it is that the larva of that brittle star is somehow able to find the coral, because it's the only place we ever find that brittle star. There's a sponge that's taken a tumble. Lots of coral rubble, some solitary coral, and this branching coral. Oh, there we go. So this is more like the goniasterid that we collected yesterday, in that it doesn't have the extended arms. It doesn't have the same color pattern. You can kind of see it wrapped a little bit around the edge of that rock. Chris suggests perhaps that it's feeding on sponge. It's also next, those little branches that are next to it are bryozoans. So we've got a mess of stuff down here on the bottom. A bunch of silica spicules of the sponge, some bryozoans growing off of that. All those little branches that you see are a series of interconnected little cavities, each of which contains a zooid that has a ring of suspension-feeding tentacles, and those tentacles actually have cilia on them that beat, so they generate their own current that comes swooping down over their mouth. So Chris says that's probably the genus Ceramaster, and therefore it's definitely feeding on sponges. He must have some evidence that that genus is known to be a spongivore. Now coming into view is this beautiful Metallogorgia melanotrichos colony. That's the sort of pinkish colored polyps that you see. And earlier today we were all kind of excited because we saw one that had what we call two heads. And by that we mean it grows on this long stalk, and all the branching is concentrated at the end of that stalk, at least in what we refer to as the adult stage. But one of the ones we saw this morning had essentially two clusters of branches, and both clusters had a brittle star on it, and that's just extremely unusual. We've seen literally hundreds, if not thousands, of these colonies, and typically have just the one brittle star. And that's going across the Pacific and the Atlantic. So again, note the difference between the rock surfaces that are raised somewhat with the flatter seafloor, and you just get a different density of suspension feeders. And it could be that the larvae of all these animals do settle on the flat seafloor, but they simply aren't getting enough food, or that seafloor isn't stable enough. If there are sediments or rubble being sort of swept across there, you can imagine that as a very small initially developing coral or initially developing sponge, you would either get buried or swept away, and so it's not a great place to live. So there are different elements to success of where things are, and one thing we have certainly noted is that the more stable the substrate is, the larger the organisms we see are, the more diverse that sessile feeding community is. See some live cup corals, some sponges, black coral, several Ovidogorgia. I'm impressed at how many of those we've seen today. This beautiful large sponge that's coming into view in the center, which I think we may have imaged a little bit earlier today. I've got my eye looking out for another one of these stock crinoids that has the snail fur growing on it. This is going to have, I think, a bunch of benthic ctenophores on the sponge. On the back side of the front of this sponge, I can see these little dots, and it looks like there are little raised structures on each end of the dot. So if you've ever seen, you can look this up, Google it, a sea gooseberry. That's a ctenophore, and the sea gooseberry is kind of the common textbook. This is what a ctenophore looks like, and it's this round, gelatinous, jelly-like zooplankton that has these two long retractable tentacles that it extends in order to feed. Well, within the phylum Ctenophora, there's one group, the platytenid ctenophores, that have taken on a benthic existence, and they've kind of compressed their body so that there's a flattened surface, and the mouth is facing down towards the surface, and they can kind of creep over the surface. But they still have their two tentacles. You just Yeah, we're going to hopefully swing around to the right here so we get a good view of these. Yeah, try to keep on the side view and not get on top of them so that we can see the tentacles from the side. So I said that the sea gooseberry has these long tentacles, and they can be retracted into what are called tentacle sheaths. And when you look at the sea gooseberry, those tentacle sheaths are basically inside the body. But with the compression of these benthic forms, there's still the tentacle sheaths, but they've remained kind of standing up above the rest of the body. So when you look in at these, you'll see that there's a long, flat body with two raised structures on either side. And you'll see these kind of white threads hanging out of them. And for most of these, it looks like the tentacles have been retracted, but they could actually extend many, 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 many times the length of the body. And it's basically like fishing line, if the fishing line were studded with glue packs, such that when a fish intercepted the line, it became glued to the tentacle, and then there's musculature inside that tentacle that allows it to be pulled back to the animal, and then it can ingest what it's caught.
That's not how you fish?
I can't get the glue packs on my fishing line.
Yeah.
It just keeps coming off.
Fisherman's not.
At least you don't get a hook in your finger that way, though.
True.
There we go. Nice view. So those are platytenid ctenophores. Yeah, so this one we zoomed in on. You see that white mass? That's the tentacle all the way retracted and compacted inside the tentacle sheath. And so, as you can imagine, if it can extend out probably hundreds of times the length of this body, it's going to look kind of globular when it's pulled in. Kind of like got a winch cable of its own, except it's not rolled up, it's compressed. And Pilot, if we could look above this one, I noticed another one, there was something, a larger red dot on the back, and I just wondered if there's, it's just maybe it's been feeding and its stomach is more engorged. I think with that, the red color you're seeing, that's the central part of the stomach. Oh yeah, there it is, the one above it. You notice it's like more globular. I'll bet it's ingested something, and that's why it looks like there's that little bump. Maybe it's ingested a little jellyfish. And the one that you see in the background, now you can see the tentacle nicely coming out of that tentacle sheath. Looks like a little chimney with a line sticking out. Very cool. Nice view. Thank you, video. Thank you, pilots. I'm also seeing there's various zoanthids growing on this sponge. You can see it through the texture of the sponge. This is a glass sponge. And this particular species of glass sponge, or this genus, is well known to harbor these symbiotic zoanthid associates. So very often when we look in, we see these polyps dotting the sponge. And to some degree, I believe there is an associate-host relationship. So it may be that these zoanthids, this particular species of zoanthid is specific to this species of sponge or this genus of sponge. I'm kind of guessing on that. I don't really know, but we see the association so much. There's got to be something more to it than just random chance. And then I'm neglecting to mention something about that euplectellid sponge. There's a beautiful vase sponge standing behind and to the left of this one. It's got its little cap dome on it. Great view.
Yeah.
Or either one. The 3D is good. So a reminder, we're scaling up a kind of a cone-like feature, and we're going up the side slope. And what you can see is that it's more like a stepped feature. So we get to a steep slope, and then we get to a bit of a gentle part, and then it's going to steepen up again, and so on and so forth as we climb. That looks like it may be another line of holes. I mean, it's certainly a line of burrows of some kind. The question is it the elongate mystery holes that we've been seeing? Certainly kind of looks like it.
I notice it's not very long.
It is not very long. Can we get
lasers on it?
Yeah. Thank you. So I do think that's about the right size, 10 centimeters. So what are we seeing? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 16. So it's about 2 meters long, almost 2 meters long, that line of holes, if you include that little curl at the top. Another sea star down below. I see a Bathypathes black coral in the distance, Enolapsamia, another one of the, I think, Candidella primnoid corals. I was just naming a bunch of things that are on this rock in the upper left. Copy that. Several of the Bramble Bamboo corals, these are the ones that can grow opportunistically either in the sediments by setting down root-like extensions at the bottom of the skeleton, or on hard substrate by modifying those roots into a kind of a holdfast, a cemented disc that it can attach to rock. I'm a little bit surprised that we're not seeing more fish on these sedimented areas. So I'm anticipating off in the distance there that that's going to be a large plate sponge. You can see the slopes kind of picking up over there. So again, we go through these patches that are pretty flat, gentle, lots of sediment, and then we get to these new rock outcrops and more rocks exposed. There's no, that's not Acanthogorgia, that's something else. Yeah, it's large. I guess it's a plate. It almost looks like it's curling over and cupping at the base. Looks more like half a bivalve, you know? Clamshell. Giant clamshell. Forward to the right, lower right of that sponge, there's a fan on the lower part of the rock, and I'm not sure what that is. Almost center bottom of the screen now. Mike Vecchione is saying he has a feeling that the hole makers require a very large, flat, sedimented area. Agreed.
Is this an Octa Coral that's kind of tipped over, or?
That's what we're trying to find out.
Looks like somebody's munching.
Yep. I think it's a Primnoid Octa Coral growing on a stony coral. Many brittle stars in its arms, and here we have, this might be Evaplasoma again, one of these corallivorous sea stars. In fact, it looks like you can see the damage it's done. Just past it, there's a whole bunch of branches extending from the coral that are denuded of any polyps. And so I would imagine that this sea star has already been out to that part of the colony. Chris Ma's comment is nom nom nom. You see the way it's kind of folded over on these corals, and it does that. The mouth is kind of pressed against the colony, and then it actually everts its stomach over those polyps, secretes some digestive juices, liquefies it, and then just sort of slurps up the liquid. What I'm liking is those bare branches up behind it, because now you can see, when I keep saying that there's an axial skeleton to these octocorals, that's what I mean. And the skeleton of these primnoids is formed largely of protein, a gorgonin, that is embedded with amorphous calcium carbonate. So this belongs to the group, the subgroup of octocorals that have a pretty stiff and hard skeleton. They're called the Calcaxonia collectively because of that amorphous calcium. And then you can see a comparison between the sea star, Asteroidea, and these brittle stars that are also on the colony, probably Ophioplinthaka, something like that. Have a lot of spines on the arm, a much smaller central disc, and the arm is set off from them. I think that the coral is in the genus Candidella. And while Chris and his friends are looking closely at these echinoderms, I'm looking closely at the coral to see if I can see the associated worm that typically lives with this Candidella, and I don't see any. It's possible I do right here. Let's see. So what I'm looking for, what I term garages. No, those are very small brittle stars. So there would be places along the branch, if you were just following the branch underneath the polyps, it would look suddenly very thick, kind of like maybe right here. And that's because the skeleton has been modified by the presence of the worm, and the coral then produces the sclerites to create a little space that the worm lives in. One nice thing about the view that you have right now, you can clearly see along the body wall of the polyp, that is the part that connects from the skeleton to the tentacles at the tip, appears to have these white rings around it. And what you're seeing are the margins of the scales that surround this. You need to think of these polyps on Primnoidae as wearing little medieval suits of armor. So that's a hallmark of a Primnoid. Really nice view. Thank you. Yeah, Chris says that the brittle stars are Ophiacanthidae, maybe the genus Ophiomitra. Sorry, folks. Was occupied there. This is an anemone. It looks like, I believe, something in the family Hormathiidae, and I'm just guessing that based on the rough texture of the outer body wall. It looks like it incorporates sand grains and other stuff. And then we have a Galatheid squat lobster.
How big is that, the anemone?
Well, let's turn on the lasers and find out. What I can see is all those dots in this sponge. Those are all zoanthids or anemones. I'm not sure which one lives on this sponge.
Wow, so that's maybe 2 centimeters across. That's
It was 2 centimeters for the anemone?
Yeah, about.
That was a lot smaller than I thought.
Yeah.
Yeah, check that out. Look at all these guys. Yeah, I cannot remember if these are anemones or zoanthids that live in regular sponge. This is what we see. These guys live together. Maybe, a few million years, this will be one organism. They've co-evolved, much like the mitochondria in our cells.
I'm loving this anemone. I'm sorry. It's just,
What do you love about it?
It's, you know what? I'm going to use a new phrase. It's alienly graceful. It's graceful in an alien, xenograce. It's just this, it just looks so strange. The way it's draped over and...
Xenograceful?
Xenograceful, yeah. Yeah.
Okay. It's gradually opening up the oral disc. It's a really great example of what a polyp is. A polyp would be that body column that is attached to the bottom, ringed by tentacles at the top, an oral disc with a mouth in the center. There's only one opening into these animals, so food goes in that opening, waste products come out of that opening.
Can we get a...
Yeah, go
I was going to say, can we get a zoomed in still, really nice, of that anemone centered up?
That was max zoom.
Yeah, nice and centered with all the tentacles and scale inside. All right, perfect. Yeah.
This yellow stuff, what am I looking at? These, I believe they're tunicates. My question is whether this is a benthic tunicate or if this is something like a salp chain that has gotten caught up on the sponge. Can't quite tell. Hermit crab's also wondering what that is. Maybe he's looking at us, wondering what we are. Here we go. Lefroyella. Lefroyella is this sponge that has all of these associates. And it's a kind of glass sponge. So now that I know the name, I'm going to see if I can do a quick search and find anything about what's associated with it. It's a beautiful view. As we look at this magnificent sponge, I'm also looking at the rock behind it. I see a couple of these little fans. I'm seeing, I think, both hydrocorals, different group of the Cnidaria, that'd be the hydrozoa. And I think I am seeing some bryozoan. The center one might be a bryozoan.
Yeah, so looking at the bathymetry again and looking at a slope map, I think we're going to come up for about another 110 meters, and we're on this steeper slope of about 35 degrees. I think this is going to be mostly carbonate encrusted talus, and then we might come up again to those slopes that we see that are more indicative of those pillow flows that might have possible exposures of 17 or 18 degrees. And so, yeah, we're going to come up another, if we come up another 70 meters, we might get there.
So this is a crinoid that we're looking at. I'm not sure that we've seen this species before. We've been seeing several of the five-armed purple morphs. This one has more arms. Again, it's a suspension feeder, getting its arms up into the water column. Tube feet on the small side branches there, flicking particles into the grooves along the arm. It's hanging on another one of the Candidella primnoid octocorals that we saw earlier. And what's right above it? Oh, that's a bryozoan. Okay. It's a bryozoan just behind it. Actually, a lot of these primnoid polyps are closed up. You can even more clearly see the scales on the polyps. There's a couple of other smaller white crinoids. It's a tough world down here. There's all kinds of stuff overgrowing each other. And so it's a little bit of a race to see who can grow the fastest and stay uncovered by your neighbor. There's a small dot to the right of the tubular sponge on the rock. It's red, if you can see the red. And it looks like a little Anthomastus colony. Yep, just 4 polyps, so that's a pretty young one. This is another kind of octocoral, has these really large polyps for the size of the body. Quite impressive. Thank you, that's good. Here again, we saw this earlier, and I was busy doing something else here in the back row, but this large sponge that is this combination of white and yellow. And I remember seeing something like this. I think it was on last year's expedition to New England Seamounts. It may have been in an earlier year along the USA Eastern Seaboard, but I know we collected some. And one of the questions was it 2 sponges or 1? And I haven't been able to follow up with the folks who looked at it to know the answer to that. So the question is the yellow an outer rind of this species, or is it literally something different that's overgrowing it? And if it is something different overgrowing it, why is it only ever overgrowing this white sponge? So many interesting questions. Huh. Very tiny recruit of an octocoral there, only 2 polyps, a little thin skeleton. Another example of recent recruitment. There's an urchin that I'm seeing coming into the screen right there. So at the risk of repeating myself, one of the reasons it's so nice to see the very, very small corals is that it tells us that, stand by, pilot, let's turn to the right on that same clump where the sponge is, excuse me, the left, I'm sorry, to the left. Between the sponge and that white fan, there's a stick. And I think that's a crinoid, a stalked crinoid with the snail fur, and I can see the branches, the arms at the top. And if it is, that's a collection target. Thank you. Oh, excellent. Thank you so much. Manuela Ramos down in Horta at the Exploration Command Center is saying that is indeed 2 species in one, Caracella and Hexadella. Hexadella would be that yellow encrusting one living on the white sponge. It's so fascinating because I see them together all the time. And so I guess my question would be, does Hexadella ever grow on anything else? And she says that the Caracella Pachystroloides has several morphological types. So there you go. So this is a live crinoid. You can see it's 5 arms at the top. It's a little bit smaller maybe than some of the others. And then below it, what you're seeing is a whole different phylum growing on this echinoderm. That's a cnidarian, specifically a hydrozoan, probably the genus Hydractina, or at least in the family Hydractinidae. And the stick that it's on is actually the stalk of the crinoid. There you can see that go all the way down. And it looks, you may not be able to tell, but it looks like it's vertebral just the same way the arms of the brittle star are. It's the same basic structures that make it up. So you might have a better sense of the size, Pilot, but it is a collection target if you think it's a size that we can get. Thank you. Something else must be maybe a worm tube, because I can see a little opening there. I don't see anything inside of it. Yeah, the base might be coming out just behind that bit of skeleton that I can see just underneath it. So, the question is how to get this. I'm not sure if this is better to pluck or to suction. Well, you could certainly do that. Yep, you could grab the base. And that's Chris up there, right? Every time. Yeah, this is going to be another crumbler, I'm afraid. It's the same thing. That would be ideal. They do use that as part of the taxonomy. Thank you. If you remember the feather stars, which are the non-stalk crinoids, they have these little feet at the base called cirri that they use to grasp the surface. And these stalk crinoids have something similar, and so being able to see that base is valuable. Copy that. There it is. There it is. That was canister 4, I believe. got the target. Just before you go, pilot, just zoom in on that white fan, please. A request from shoreside. All right, great. Yeah, I still believe this to be a bryozoan. Let's see. Or is it? Well, now I'm a little less confident. Yeah, that's a tough one. It's really small. Okay, Asako agrees that it's bryozoan. I was starting to doubt myself, Asako, that might be a hydrozoan. The problem is there were no tentacles and or lophophores extended, so it's a little bit tough to see. It otherwise had all the structure of the bryozoans. That little polyp in front of it, I think, is a bamboo coral, which is kind of cool. A little baby bamboo. All right, let's see. Yeah. There's just no feeding structures extended, which is really not helpful. But I don't remember these little spine-like features on the bryozoan, which is making me think maybe this is Octa Coral. If it was a bryozoan, I should be seeing a series of cases with holes that the lophophores would extend out of. That's about as good as an image as we're going to get, so I think that's good. We can preserve that, send it to some colleagues, and get their opinions on what that is. Thank you. Well, I don't like it as a sponge. It seems too hard. I don't believe it's a carnivorous sponge. Oh, I'm sorry. Thank you, Caitlin. Yeah, well, maybe that larva made a bad choice of where to settle. It's growing and it'll eventually make its way out of there, right? I'll tell you one thing, though. It's a pretty good size, so it can't be that bad a spot. One good thing about being underneath the rock is that it's not getting covered in sediments, and so it's not getting buried. Yeah. Oh, I'm kind of interested in this. Yeah, I don't know what's underneath the rock. Oh, up here is a Nemerdian worm? No. Or no, is that a hydroid? There's just too many things to look at in this small scale. Yeah, that's what I'm looking at right now. This worm thing. But I can't identify it. It's just under the sponge. Yeah, you can see some pink insides. Not much to be said about that one. It's too small. Yeah, so that thing, what's that? It does. Almost like the, but I don't, or is it a tube foot? Or is it a worm? Or is it a, insert your guess here. Guess from Tina Sacco is a turbularian worm, which would be a flatworm, platyhelminth. And both of those could have been turbularians. See another Metallogorgia in the distance. Leopathes off to the left. Some more of the Candidella primnoids. There's two of them here. Some yellow encrusting sponge that we had identified earlier. And still, dominant on the bottom is all this coral rubble. but most of that coral rubble here is dead. I'm very curious to see if we'll see any alive up top, or if this is a reflection of something that was going on here 20 to 40,000 years ago, and these are just the remnant skeletons. This is entirely possible.
And if you are just joining us, we are about 50 kilometers west of the island of Flores, about 170 kilometers from the mid-Atlantic Ridge main axial valley. We're diving to a depth originally of about 1,500 meters below sea level, and we've been slowly coming up one of these outer ridges on the outer flanks of this Guyot. We're currently at a depth of 1,445 meters below sea level, and we might come up another 50 to 100 meters in depth throughout the course of the dive. We're on crust that's about 13.5 million years old that was formed through mid-ocean ridge volcanism at the mid-Atlantic Ridge. We've been diving on the mid-Atlantic Ridge for the last 2 weeks. Mid-Atlantic Ridge is an example of a divergent plate boundary where new oceanic crust is generated and the tectonic plates move away from each other. We're currently on the North American plate, and the Mid-Atlantic Ridge to our east separates us from the Eurasian plate. Now we're starting to get into an area south of here that's interesting in that it is a junction between two segments of the Mid-Atlantic Ridge and then another ridge, the Tercera Ridge. And that's what we know, what we call a ridge-ridge-ridge triple junction. It separates three tectonic plates. It separates the North American plate from the Eurasian plate, which we've been diving for the last 2 weeks. And then when we get into this triple junction area, it also separates the African plate. Now, overprinting all of that is this large bathymetric high, this underwater topographic high that we call the Azores Plateau. And that's that's formed through a volcanic process known as hotspot volcanism. So we have a deep source of mantle material upwelling right near this triple junction. And so it's kind of a confounding level of factors. So the oceanic crust that we're sitting on that was generated from mid-ocean ridge volcanism at the mid-Atlantic Ridge is, as I said, about 13.5 million years old. But overprinting that is much younger volcanism from hotspot volcanism. That hotspot volcanism formed all the Azorean islands. The island of Flores, for example, is about 2.2 million years old, and some of its oldest volcanic products. That was the volcanic products that were formed during the underwater eruption. We're just a little bit west of that. We're just about 50 kilometers west. And, we're probably in a similar age range, 2 to 3 million years old in terms of the volcanic products that form the base of this Guyot. This Guyot has been covered in carbonate from dying coral, dead coral, also marine detritus that's fallen through the water column. And as that material consolidates and remineralizes, it forms a lot of this darker crust that you see. You can see right now some of the skeletal material that looks like a gnarly surface in between and beneath some of that black crust. Some of that black crust is this concreted carbonate material, and some of it is also older pillow basalt flows. Overprinting all that is some amount of precipitation of minerals like iron and manganese. And hopefully as we come up to this steeper end, we'll get a little bit more exposure, possibly of some of those basalts we saw earlier, and maybe even some more pillow volcanoes. These pillow volcanoes, which you saw some really good examples of in a dive 2 dives ago, tend to be kind of overprinted or kind of buried with all this marine sediment. Now our ROV team is just going through an ROV pilot swap, so they're doing their exchange of information. So we're kind of just hanging out here for a minute or two while they do that handoff. See maybe one lone Octa Coral right in the middle of the screen. All right. Thank you. Which one are you going to pick, Scott?
How about, so you see the yellow fan with the white sponge at its base. Just to its left, I see like a puff of branches, kind of a ball-like shape. Let's go down and look at that surface. See what's in there. No, to the left of that. It's almost directly in the center of the screen.
The pink pom-pom.
Yep, the little pom-pom with the fan next to it. Let's see. So this is a Chrysogorgia. It does have, I imagine it's either a shrimp or a squat lobster. Probably a shrimp based on an earlier collection. That bright lemon yellow thing up there is a sponge, maybe Hertwiggia. Yeah, so we've seen many examples of this family today, the Chrysogorgiidae, and we've seen lots of colonies of Chrysogorgia. I really have no idea what genus this is. Oh, I can see at the base there, that's actually a squat lobster. I just see its bright, its eye reflecting the light of the sub, and I can see one of its long chelate arms, clawed arms, extending downwards. So maybe that's what the other pink thing that we see in here is. Oh yeah, absolutely, because now I can see coming up above it, there are two very long arms. So this looks like a pair of squat lobsters just taking over this Chrysogorgia as its home. And just reminding you again, the name means golden coral. You can kind of see the coral through the tissue. And then this is a bamboo coral just next to it on the left. And where's this one branching, Ashton?
I think it's branching at the node.
You are correct. There's a nodal brancher. I'm going to guess probably a relatively young colony of something related to Jasonisis, what we call the J3 clade.
I knew that. You could have asked me that as well.
Yep.
Yeah.
I think that might be all that we've seen today, actually. All right, looks good. And I just had an email from Mary Wixton at Texas A&M University, that crustacean that we were just looking at, or on the Chrysogorgia colony that had the two crab-like animals in there. And she says those would be in the genus Uropticus, and that genus is known to associate with Chrysogorgiids, and she notes specifically those very long and strong claw-like arms. Thank you, Mary. Again, the power of telepresence. We have scientists from all around the world who are watching this stream along with you at home and sending in their observations either by email, through a chat room. Some make observations on Twitter and Instagram. A lot of different ways to transmit information to us. So again, in the midst of these relatively flat areas, we come to these local topographic highs. And every time we get to the topographic high, the abundance and diversity of suspension feeders increases. And this little rugosity and change in the surface alters the regular flow. And that's probably why these animals are congregated here. See some Leopathes black coral, very tall columnar Chrysogorgia species. Some of the Candidella, 2 or 3 different kinds of sponge, and Acanthogorgia, things that we've been seeing all day and we've been looking at in different views. That thread-legged shrimp climbing around on the bottom. I'm not sure, Pilot, if we've seen the sponge that's in the upper left corner. You see it's bright white sticking out of a more yellow golden base. So I'm not sure. Yeah, I'm not sure if that's new. It's possible that, I don't know, is this an earlier stage of the yellow encrusting sponge overgrowing the white sponge? I have to look at what Manuela identified these as earlier. Yeah, earlier she had said Caracella and Hexadella, and that was this Caracella was this massive globular morphology. So I don't think that's the same. That looks more like the Lefroyella. So getting a look at the back of this sponge, and if that is a Candidella, and I think it is, then that one's almost assuredly got polychaetes associated with it. And I'm referring now to the branching coral that's sticking out from the front side of the sponge. I'm looking at, some areas where the branches are clearly very swollen relative to other parts. I don't know that I've ever actually seen the worm on a video. It's easy to see it when you have collections and see them in a dish. Just a reminder that sponges are basically plumbers, and they channel water in through the body, and they have a series of pipes that get smaller and smaller and smaller until they get to these little chambers. They're called choanocyte chambers. And lining the chambers, kind of like wallpaper, are a series of cells that have flagella on them, and they're all beating, and that is what creates the current that sucks the water into the sponge. And each of those little cells has extensions of the cell membrane that form a little net, if you will, that capture the bacteria and other particles that are coming past that cell. So that's how they're feeding. So I would like to suggest that maybe we take a chunk of the yellow sponge. So what's your situation? Can you roll around to the left, or do you have to roll around to the right, or come over the top? I'm not sure. Yeah. Well, I think that would be better if you can arrange that. Sure. I agree. I don't see that Manuela is still in the chat room. We could verify for me whether this is the same or a different sponge. It looks kind of different to me, and that's why I'm suggesting a collection. But I'm going to keep my eye on that chat room. Or Manuela, if you are in the ECC and you're connected by the phone, then feel free just to get on the phone line and say something. Yeah. We want a piece, so I think we have still 3 bio boxes open, so that could be used. Doesn't have to be suctioned.
You learned an interesting sponge fact today, didn't you? We both did. When we were reviewing.
Which one?
The continental sponge silica update.
Oh yeah, I did notice that. Yeah, thank you. So yesterday we were talking. Yesterday, the day before, a couple of days. They're all blurring together. We've been talking a lot about silica, and Ashton's been talking a lot about silica in the rock and in these pillows, and I was noting that the sponges are largely silica-based, all of these skeletons that they're making, and we were noting that there's an interesting, the circle of life. There's a transition and a movement of silica molecules between the rocks and the water and back to the sponge. And Ashton showed me this paper that was doing some estimates to understand what are the pools of silica, where are they? And in the water column of the dissolved silica that's in the water, up to 36% of that is used by sponges, at least in this estimate. They were looking at sponges on the continental slope. 36% of that silica is taken up by sponges for their skeleton.
Yeah, it's a huge number.
That's incredible.
Yeah. And then the rest of it, at least in that estimate, is largely buried through diatoms. But it brings up an interesting question of, maybe there just isn't the data to do those estimate for deep water sponges or sponges off of the continental shelves. Now, most sponges do have a silica structure. That
is correct?
But there are some calcareous sponges.
There are some sponges that have calcareous spicules. Those ones, they also have silica.
Okay.
So very few sponges have the calcium. Virtually all sponges have the silica. The ones that have the calcium also have the silica hard parts.
Have we seen any of those?
No, they're a pretty restricted group. Yeah. I'm not sure I could recognize one if I saw one. I'd need a sponge specialist to tell me if there was such a thing.
We'll have a sponge specialist on our next cruise.
We will. You don't need to restrict yourself to the yellow sponge, so you're sort of going for what's an easy target. The white sponge is most certainly different from the yellow one, so I'm not necessarily looking for the white one. In fact, I think we may have collected that white one a couple of days ago. But yeah, if there's anything else, okay. This may be a lot smaller than I thought. You may end up having to suction it anyway. Yeah. Let's see what happens when you grab it. Because I don't know the texture of this, Andy. One might even say that's spongy. All right, that's great. So we got a chunk of that sponge in the port outer bio box. Oh, was that directed to me? Okay. Come on. Yeah, absolutely. So sponges, because they are sessile, well, most of these organisms we're looking at are sessile, meaning they're attached to the substrate. They can't run away when a predator comes, and organisms in that status usually have some kind of mechanism for protecting themselves. And especially when we see these sponges that have a lot of different pigments to them, that is, color to them, they have what we would call secondary metabolites, various kinds of biochemicals that may be noxious or toxic to other organisms. And some of those may have some biomedical benefit to us. So, for example, they may serve as an antibiotic. And so there's a lot of interest in what are the compounds that sponges are making, how might we as humans benefit if we could synthesize those same compounds ourselves. So it's a whole field of natural products chemistry.
Here's some sand stripes.
Yeah, I'm looking at them, trying to see if they're... I don't think they're the same.
Oh, yeah, James Aubrey, my grad student. Oh, is that right now? Like how early relative to the collection?
Like 30 seconds ago, but it didn't look the same to me.
Yeah. Oh, they're the same. Look at that.
Oh, were those the ones we went over?
That's the
downward.
That's the downward
yeah.
Zeus 2? What Mini Zeus?
Mini Zeus.
Mini Zeus. So let's make a note of that.
Can't get away from these holes.
Yep. Thank you, Caitlin. Caitlin just noted on our, I'll call it brow-mounted. I don't know if that's, I'm sure that's not the right word, but what do you call that pilot, where the mini Zeus is? The brow? Oh, great. Okay, so the brow, so the upper bar above the main camera we're using, there's another camera that's pointed more straight down so we can get a different angle of the sediment. And here we go again. Here's two more of these rows of holes coming just to the right of the lasers. He is. Yep.
I'll put it up on stream 2 on HD2.
If you check over on camera 2, you can see a view from that mini Zeus camera, so you can see much better looking directly down at these lines of holes, and there's 2 or 3 of them right here. And the one with the lasers on it right now looks pretty fresh and actually relatively short, I would say, compared to the others. So maybe that's a sign of something. I thought you made a really good comment earlier that already we have started making assumptions about this. And that assumption is that it's formed in a linear progression. We don't know that.
Yeah, it just so happened that the first ones we saw were these nice, beautiful, long lines. But now that we have a N of 5 instead of an N of 1, we see that some of them are short. They seem to have some curvature occasionally.
These look fresher than the ones that we saw this morning. Anyway. So we're zooming in on camera 2 with the mini Zeus to have a look at these holes, see if we can see anything in them.
So Michael is asking again if there's any way that we get eDNA from near the holes. I think we fired off our...
Yeah, I think we, well, yeah, we only have one left, I think.
Which is the bottom
is that
one.
Right? No, we should have 2 left.
Yeah, we have a bottom and a DSL.
I inadvertently crossed off the wrong thing. I meant to cross off the canister. So, so Mike's suggesting that we take an eDNA sample in the water. Is that what you mean, Mike?
Yeah.
Sure. Yeah, that's fine with me. Pilots, yeah, let's fire the Niskin 4, please. Thank you. Science is ready. Sample collected. Thank you. Boy, that is really angular. Those are very, very fresh.
weight here.
Right, yeah. So Brian, one of the video engineers, is saying that it's almost pyramid or triangular shape. Like, I think you mean like they're wider at one end than the other. Yeah, so it's not like a flat paddle, which is kind of interesting. So Mike was talking earlier about a suggestion made by Les Watling that there's a type of shrimp that could do it. And so you could imagine if something's digging a burrow, it's going to be wider at the front perhaps than the back. Or vice versa. I'm trying to think of the way my dogs dig into the beach, you know, and I guess it depends how clean they burrow. Yeah, these ones are mostly infilled. So those ones look pretty old. All right, I don't see anything sticking out of any of these holes, even though I see it fresh. Yeah, that looks like one of the thread-legged shrimp. Yeah, maybe if we could, can we zoom in on the main Zeus? Thanks.
If he's digging a hole.
Yeah, exactly. If I think it's the thread-legged shrimp, in which case those legs are way too thin for digging holes. Yep. Yeah, that guy is not going to be digging any holes. Interestingly, there's a hermit crab right beside it with, I think, an anemone on its back. No, I don't think the hermit crab would do that. Hermit crab's got a home. Oh, what's that, though? Well, now that you're down here, that's another bryozoan. What do you know? This place is bryozoan crazy. I've never seen so many bryozoans that I could identify as bryozoans. That's very interesting. And actually, there's a isopod I saw stuck in a little shell up there on the left. Okay, I think we've kind of exhausted what we can do here. Thank you. Just before we saw these holes, I was going to relay to you some information that my grad student James Aubrey is a PhD student at University of Louisiana at Lafayette, and he's working on the Acanthogorgiids, the crown of thorns Octa Corals. And we had been talking about how sponges create or produce these compounds that might have medical benefit. And he notes that some corals also have compounds that may have medical applications. And in particular, again, some of these really colorful ones that have these pigments may be of a lot of interest. And some of the shallow water ones have actually been had compounds that have been brought to market. But as I understand it, I was doing a little bit of research on this at the beginning of the expedition. Most of the corals that have been examined for that use have not yielded anything that could be brought to market commercially. So there are some examples, but most of them fail for one reason or another. I don't know if that's because it's too difficult to synthesize or they're not useful in a broad enough context. So it seems like the sponges have more interesting products than the corals. Thank you, James. There's a very nice review on these sorts of questions by Dr. Paul Snellgrove, who's at Memorial University in Newfoundland. Right below, let's see, to the 5 o'clock of the lasers, almost at the bottom of the screen now. There's something light beige, branching, not the tubular thing, but below that. Yep. Might be another of these bryozoans. It's directly right of the lasers, halfway between the lasers and the edge of the screen. they blend in really well. And actually, I'm seeing several of them now, which makes me wonder, you know, how many have we passed over? They're sort of disguised here on the bottom. It's just nice to get a view to, again, just remind us of how much is down here. Yep. Yep. Taller one. Yep. Yeah, interesting. So this is, again, this bryozoan. You can actually see it much better. And note these little rings of tentacles. It's called a lophophore. Those are the ones that are made up of probably a couple of dozen very slender ciliated tentacles. And the ciliated means that there are little hairs on those tentacles that beat, and they create a very characteristic current for feeding. Thank you. And then let's look at the top of this colony. There's an anemone, I think, growing on there. There we go. It's kind of an interesting one. Is that an anemone? Or is that a nudibranch? That is Maxim? Thank you. Yeah, I'm not... Interesting. I am not 100% convinced that that is an anemone. Well, we'll see if someone on shore recognizes that. Thank you. Interesting. I've said it before, I'll say it again, you just never know what you're going to see when you zoom in on that seafloor. No, thank you, pilot. We can move on. Thanks. Far upper right corner, there's a white wiry coral. Maybe we could have a look at that as you get up there. Thank you. I'm pretty sure, I'll check with Nav on this, but I'm pretty sure we're probably in the last 40 minutes or so of bottom time. Thank you, Nav. I have confirmed that, or Nav has confirmed that for me. Oh. Should have collected that bryozoan. I think that's a cup coral. Yep, it looks very white underneath, kind of stiff. Just has that appearance, you know. I think bird watchers and butterfly watchers kind of know this. If you looked at enough birds, you start to get a feeling, even subconsciously, you know what the bird is before you can clearly identify it. You know what group it's in. And that cup coral had that feel for me. I'm now regretting that I didn't ask you to collect that bryozoan with the whatever was on it. Perhaps. Let's look at this white thing first. We haven't gone very far, but otherwise I think we should get a bryozoan. There's so many bryozoans. They're so rarely collected. I think it would be a missed opportunity. Okay, so we have seen this before. This looks like a swiftia to me. white one. It's a type of plexaurid. This is an Octa Coral. All of the polyps have been withdrawn inside that tissue, and that's why you only see these little humps along the skeleton. And there's one lone amphipod sitting on one of the branches. Thank you. That's great. Yep. Go ahead, Nev. 3030. Thank you. 30 minutes. Yeah, so maybe pilot in that case. I'm not sure how far back that was, but we're seeing several other fans, bryozoan fans in front of us. The reason I liked that other one was that thing that was on it. Okay. All right. Okay. Yep, it's right there. Okay. Yeah, video's just showing me the replay, so you're certainly going in the right direction. It's just a little past and back from this Leopathes. You're really close to it now. Might be under the skid. Sure. So it was that one, right, Caitlin? Yeah. I think it might be right here, huh? Yeah. So it's straight in, it is in view now. You don't have to go back any further. Yep. It's between the lasers are almost on it now, just a little more left. Now it's above the lasers, right between them. Yep. Thank Caitlin.
Caitlin rocks.
So I'm seeing, again, this is pretty small. So what do you think, Pilot, about trying to suction this by scraping it from the bottom? Yeah, let's give that a shot. And I think that's probably our best chance to get what's on it. And if that's an anemone, it's going to be stuck to it. But if it is some kind of nudibranch, it would release if we tried to grab it. Yeah, we have one jar left, I believe. One canister left, number 5. And I think we may have rotated the canisters to 6, but 5 should be empty.
I think you're right, Scott. I think nudibranch.
Yep. Oh, yeah, you think nudibranch as well? Cool. It's something about, you know, if those were tentacles, I'm not sure where the mouth would be. There is a pom-pom anemone that has tentacles kind of like that, but it looks like it has guts. And anemones don't. Yeah. And pilots, right now, I think you're on canister 6, but that was for a blowout. Canister 5 is empty. If you're interested in seeing different views of this collection instead of just this main view behind the section, you can turn over to channel 3, where the quad screen will show you 4 different cameras that the pilots are using so they can see various angles in order to position the nozzle as best as possible. All right, I think I heard it went into the canister. Oh, yeah, no, we want to try to keep scraping. Yes, please. Huh. Boy, it's really on there good, isn't it? Right there. Oh, you must have got it this time. Wow. That's incredible. I really thought that as you scrape the bottom, it would just break off, but it's amazing. Yeah. Sure. So I think you were trying to like scrape the nozzle on the rock, right? Yeah. Yeah. I guess that's what we have to do. Thank you. Pilot, is it possible to turn off the suction while you're doing this? Yeah, just so the nudibranch doesn't keep getting vortexed around in there. I saw it go down below. They just moved it. It's good, I guess. It doesn't you need to rotate the canister back to 5? Okay, thank you. Understood. There. Yep, that's good. Pilot, I apologize for ever saying that anything is in an easy location to collect. All right, folks, we have about 15 minutes left on Cachalote Seamount, or Cachalote Guyot. For those of you who weren't here for our morning briefing, I learned last night that Cachalote, and I apologize if I'm mispronouncing that, it's a Portuguese word, means sperm whale. So I know what I'll be doing as I'm waiting for the D2 to get back on deck. We've had no reports, though, during the day of any sperm whales in the area. I see a crinoid down there. Not quite as much fauna right now. Can we have the lasers on as we transit here? Maybe we can zoom. There's a sea star right at the center, just below the lasers now. It's almost at the bottom of the screen, still in the center. Bottom of the frame, excuse me. Yep. It looks like what I would remember hearing called something along the lines of a Henricia. Lots of arms and not much to the central disc. That's the one I'm talking about. All the way at the bottom is a crinoid. It's the white one to the top, right? In the center. Okay, thank you. Nice. And just in anticipation, I will let Chris know that we've already filled all the suction canisters. Oh, thank you. Jean-Marc Gagnon notes that it's the same word in French, cachalot, for a sperm whale. That's cool. And I know that the asteroid biologists use the pattern of the ossicles and the spines and to some degree the number of rays, arms. That's good. Let's see what else we can see here in our last 10 minutes or so.
Hey, Scott.
Hi, Chris. What did you think?
Hey. Oh, it's actually good timing, or a good observation. So this is what's called neomorphaster. We have a lot of these on the west coast of the United States, and there's another species that occurs on the European side. The one we have is Forcipulatus, and the one on the European side is, I'm blanking on the name. On our side of the coast, it's pretty common. So it's actually something that I wouldn't have asked to collect unless, we had like plenty of time and space, which we don't. These are actually kind of cool just in their own way because these are what's called sticasterids, and so these are related to like the common Asterias and Pisaster, but they're a very early like diversification branch of sea stars that occurs mostly in the southern hemisphere. There's only a couple of them that occur in the northern hemisphere, and they live in the deep water, and this is one of them. There are fossils similar to this animal in the Cretaceous of California and other places in North America. So, it's one of these animals that takes on kind of a living fossil appearance. It looks very similar to something that occurred in the fossil record. There's a lot of animals at this depth that are kind of presenting that look. It's always been a question as to whether or not, any of these, as to what they're doing down here, sediment feeders or what have you. So it's always, ecology and trying to, and helping out a lot of my paleontological colleagues with what their fossils are doing with living examples is always a fun exercise. There's one of the things that I've been noticing at least 3 or 4 times is we're seeing dramatic range extensions on this cruise. That is to say, we have several species that we kind of have some familiarity with, but none of them have ever been shown to occur this far out away from the coast. And of course, that's because we haven't really looked that closely at all of these species, or at least don't have. If there have been collections, we haven't really been able to document them yet.
Chris, can I interrupt you for a second there?
There's this remarkable blue thing.
Yeah, exactly. I just wanted to interrupt you just to be able to say I'm pretty sure that this...
Yeah, please go ahead.
Okay. I said it was a tunicate. Actually, I changed my mind. I think it's actually a sponge. It can't be a tunicate because there's no organs or zooids inside. So that's a sponge, beautiful blue color. I haven't seen this one before. I noticed it from a distance. It was really glowing in there. And clambering up behind it, I believe, is an isopod. Cool stuff. That's good, pilot. Thank you.
Thanks, Scott.
I don't want to wait for the...
I know we're put down to the wire.
Thank you, Chris. I don't know that isopod's going to come out, so let's use the last couple of minutes. That was a really nice find, that sponge here towards the end of the dive. We haven't seen one all day. Quite unique, and I wonder if they're often tucked into those little crevices. Looks like maybe a Hylanema type sponge. I'm seeing more pillows here.
Yeah, at least the outer rinds of them. Is that a fish right there?
Fish.
Or is that, no, that's an Octa Coral. Left? Yep.
Yeah, that's probably a Chrysogorgia Octa Coral. See a yellow Enelopsamia coming in at the top. Slope is really picking up here. Looks like there's a lot of stuff coming in above. So it's interesting, today we did not get to a point where we saw lots of this live, maybe it's Selenosmilia, the stony coral, the branching stony coral that's producing all of this rubble. We did see small colonies of it. Unless they're all colonies of this Enelopsamia, that's quite possible. But even that, we haven't seen huge numbers. And that just suggests to me that this is a representation of something that's been going on in this environment for a very long period of time, probably thousands of years. Now, there's some really interesting information that Asako Matsumoto is passing on. So she has a colleague, friend, she's passing this on from Tina Moltsova, a friend who studies bryozoans, and they said that bryozoans have very small and so short-lived larvae that have very short ranges. That is, the larvae don't spend... let's have a look at the sea star that's on the left side coming into view of the rock. It looks like it's in a feeding position. So the larvae don't survive for very long, so they can't travel very far. And she says they can't get from one seamount to another. And he had real trouble finding the same species of bryozoan on adjacent seamounts. They were closely related, but not the same species. So that suggests that they've become isolated, genetically isolated from one another because of the short distance that the larvae travel, and so speciated. So there's a hidden diversity of bryozoans that's probably underestimated, because we may make this assumption that, the one we see here in the central Atlantic is going to be all over the seamounts around here. But if the larvae can't get there, that's not the case. So that's really interesting. Somebody's going to be really excited any second now by this observation.
Oh, I'm here.
There you are. Yeah, this is the first of this, Chris, and go for it.
Oh, well, this is, I think I mentioned earlier there were some hallmark species that I was really hoping we would see, and this is one of them. I think I had mentioned earlier that there were some species that, by virtue of their rarity, they were sometimes the only, members of that particular group that we had ever seen. And, Evoplasoma, for example, was known only from a couple of known from only half a dozen specimens, and each one was a species. This is one of those things. This is Mixaster, and probably in the Mixasteridae. Up to a point, these were extremely mysterious. They are kind of related to pterasterids and slime stars. They occur all throughout the deep sea at this incredible depth, 1 to 3,000 meters. They're kind of reminiscent of things like pteraster, because as we draw in close, you'll notice that there's an osculum right in the center of the disc. So, we didn't know what they were doing down here for quite a long time. And I think recent video and, literally within the last year or two, people have been recording these feeding on anything from other sea stars to Octa Corals. And, and they're, oh, wow, that's beautiful. And, to add more mystery, when we sequenced them and we looked at them in their family tree of asteroids, they occur down at the bottom along with pterasteroids. And, bottom, I'm not going to get into the phylogenetic stuff, but basically that means that these are considered sort of an offshoot early on, or not early on, but, at the base of the tree. So they're of interest to people looking at the early origins of sea stars. And there are Jurassic fossils of animals that are very similar to this. So, and I'm literally still trying to figure out how many species there are of this animal, because some of the early material is just so badly mauled by trawls and whatnot. So, the fact that we're seeing them alive, is now more common than it was a couple of years ago. But I remember the first time, these were seen, people misidentified them because they don't, they weren't the things that people were most familiar with. Because the only people that had ever seen these up to that point were a handful of taxonomists. Maybe 2 or 3 people in the world even knew what they were, much less, had ever seen one. So, and so now I see these almost every time we do a deep sea dive. Either Mixaster or Asthenactus. And Mixaster is the one that occurs the most commonly in the Atlantic. There are some fine details there, but but it's always a pleasure to see one. And usually when we see them, they're doing something interesting. So I'm always looking forward to the next observation. Oh, and we
thanks, Chris. Yeah, I have to interrupt you because we're just about to end the dive, and so I just wanted to get this last view. We had a request for this. This looks like an Anthomastus.
I'll talk to you later. Bye-bye. Thank you.
Thank you, Chris. An Anthomastus, and next to it maybe a single polyp. I don't know if that's Anthomastus or something else, but again, it's a recruit. So exciting to see that. And behind it, we see a bryozoan. So this is a fantastic way to end this dive. We just saw that brilliant blue-purple sponge, the first time we've seen that. Chris just described that sea star, first time that we've seen that. And here's this beautiful Anthomastus and a single polyp recruit next to it, which tells us there's ongoing recruitment. Thread-legged shrimp coming in to say, that's all, folks, for the seafloor part of our dive. Terrific. One more thing I just wanted to thank. Bjorn Burning is the bryozoan biologist who passed on that information about the short-lived larvae, so that's great. Thank you to all of our scientists who joined us in the chat room today and on the phone, providing information, exploring along with us. Thanks to you at home who have been following along on the internet or live stream. This is the end of our dive to Cachalote Guyot. We started at a depth of, let me check my notes, 1,498 meters. We are currently at 1,425 meters, and we're about 50 kilometers west of Flores Island. We'll be going up into the water column, get one more DNA sample up in the water column, get on deck, and then look after our samples. And then we'll be back for our final dive of this expedition, Voyage to the Ridge 2, tomorrow at Gigante, I believe. Gigante Seamount. And I don't remember the depth. So I think it's similar to what we're doing right now, but check out the website. It'll give you some information. We'll be back at, 6:30 a.m. East Coast time, United States, 10:30 a.m. UTC, and you can figure out your own time zone, what time that is for you. So folks on shore, if you're going to join the pre-dive, what do we call that? The dive planning call? Losing my words. Give us about 7 minutes to just stretch our legs a little bit. Thank you. Oh yes, and we can't forget the mystery holes. We saw lots of observations of those today. Don't know that we're any closer to an answer, but we did our best. Thanks everybody. This is Scott France and Ashton Flinders signing off for the day.